I'm from Turkey. I'm from Gazi University Medical Faculty, Microbiology Department. And first of all, I would like to thank Johan David especially for this excellent organizing. And many thanks to local organizing committee and especially students, all students. And another thank to university management and all sponsors. I'm very happy to be here in this beautiful Bogota, and I'm very happy to be here and with you to discuss and to learn about blastocystis. And during the workshop, we have also discussed many things about blastocystis. I think we also continue this job for a long time. And all of us agree to more investigations and more collaborations at the end of the workshop. And so this afternoon, we can start first session with Kevin Tan. And he is associate professor in Department of Microbiology, National University of Singapore. This year, on August, he had organized blastocystis session in the first time in ICOPA. And thank you, Kevin, so much for this facility. And I think you know him very well because he has many useful publications, especially I know we have many younger researchers and the students, and they are beginner of this job. So I suggested his publications so much, especially reviews. And I know I would like to invite him for his talk about new insights in blastocystis molecular and cell biology, new tools, animal models, and implications for microbiome studies. His also research interests, especially blastocystis cell and molecular biology. So, Kevin Tan, please, for your talk.
Thank you very much, Funda. It's such a pleasure to be here. And let me first start by saying a very big thank you to the organizing committee of the second IBC. Juan David, Funda, Rune, you've done an amazing job. This is, as Rune said just now, this is a dream come true for many of us who work on blastocystis. We never thought the day will come when we will have a conference on one parasite. So we have brought many exciting talks to you, and we are very glad to see such interested participants. And we promise you that if you ask us something, we will try our best to help you. We want the field to emerge, to do well, and to keep going. Okay, so thank you very much, and yeah, I've really enjoyed Bogota. It's my first time to South America. I've enjoyed your hospitality, your food, and your culture. I think my microbiome is much better now. It's a Bogotan microbiome, and I feel healthier and younger. So it's the right place to be. I'm going to tell you three stories today, three very recent kind of projects that have been going on in my laboratory. None of it is published, so I thought it would be interesting to share with you what is happening in my lab. We haven't, for some projects, they are not near publication level. For some, they are, so I just want to share with you. The work is mainly supported by a Ministry of Education Tier 1 grant. So just to acknowledge them. I like to put this on my slides because every time I give a talk on blastocystis in a country, I will put it on the map. And today, I am in Bogota, Colombia. So I have traveled the world trying to talk about blastocystis since 2003 in Hawaii, right? So it's been 15 years of travels, and everywhere I go, and I'm sure Funda and Rune and the rest do it too, we try to spread the research interest on this parasite. So I'm very happy to be here for the first time talking about recent data. So today I will give you a bit of history of Blastocystis. It's good to appreciate the past before we can work on the future. 3 projects, one on cell biology, one on parasite-host interactions, and a last one, an interesting project that has implications on how we look at the microbiome, in particular how the parasite would interact with members of the microbiota. Let's start from the beginning. We all talk about this paper, Alexief, 1911. Have you seen the paper? This is the paper, okay? I managed to dig it up from the Singapore library. If you want the PDF, I have both the PDF of Alexief and Brahm's paper. This is only the cover, but I want to show you how beautiful the drawings were over 100 years ago. Alexief already drew what we know as vacuolar forms, dividing forms, and what he thought were cysts or multiple fission forms of the parasite. Now, at that time, he didn't know it was a distinct parasite. He thought it was a cyst of Trichomonas. Okay, so at that time, they didn't have molecular genetics to tell parasites apart. 1 year later, Bromt published in the Society of Exotic Diseases, and he managed to get Blastocystis from the stools of a diarrhea patient. And he drew these very beautiful pictures. Again, you see the typical vacuolar forms, polar organization of the nucleus or nuclei, and then binary fission. Then he said that when this patient recovered, he took the stools again, and the parasites looked different. They had become a bit smaller. So already in the early days, they had already noticed some differences between diarrhea samples and samples from formed stools. So some interesting history, and already in those days, we noticed unusual multinucleate structures and all that, and that could have led to some confusion as to the cell cycle of this parasite. This was the first time that it was called Blastocystis hominis in 1912, and the name stuck on for many years until in 2007 when Rune published a very nice review that put a consensus on how we should name Blastocystis. We no longer use hominis, we use Blastocystis species subtype 1 to whatever, all right? So today we don't draw, we use the microscope, okay? And we can appreciate that Blastocystis is an extremely pleomorphic organism. In cultures, they don't just exist as vacuolar forms, they're actually quite heterogeneous. You can find big vacuolar forms, smaller vacuolar forms, fecal cysts, and unusual forms. We're not sure what these are. You have your granular forms and what appears to be forms that are amoeboid in nature. From the electron microscope, if you do a transmission electron micrograph, they are very pretty. They have a large central vacuole of unknown function. Okay, we don't know what this vacuole is for. Probably something rather important because it's almost always there and takes up about 90% of the cell's volume. And because it does that, organelles are pushed to to the periphery of the parasite, where you find the nucleus and the mitochondria within the cytoplasmic rim. If you look closely, most parasites' nuclei would have a karyosome mass, a darker staining region. It's quite typical of Blastocystis, and you find mitochondria closely associated with the nucleus. Can you tell which is the host that we obtained this parasite from? If you know, this is one of my first papers with Prof. Yoshikawa. It's from a cockroach. This is cockroach Blastocystis. Right, so even cockroach has Blastocystis. And when Prof. Yoshikawa did the sequencing, it was very different in terms of its ribosomal RNA. It was an outgroup when you compare it to the other Blastocystis. So already in that time, we had parasites that really look, you can't tell them apart, okay? So I'd like to start with my first story, the first project that was done in collaboration with Professor Cynthia He and our joint postdoc, Dr. Lee Feng Jin, and it has to do with the parasite cell cycle. Okay. As mentioned, and for many of us who work on blastocystis, whether in exenic or non-exenic cultures, you'll find this heterogeneity in the cultures. There's amazing size variations and nuclear variations as well. Sometimes you find 1, 2, 3, or 6 nuclei. It's unusual because many parasites don't exhibit such heterogeneity in size and in nuclear numbers. Okay, so even in this culture, you can see cells that are single uninucleate or binucleate, and the size is quite variable. Now, my feeling is because of that, it has led to many postulations on how the parasite divides. So already in 1991, a very well-known blastocystis researcher from the US, Charles H. Ziert, published his idea of how the parasite divides. He proposed 4 modes of reproduction: binary fission, which we observe quite regularly in culture; plasmatomy, where there's an asymmetrical division of cells; schizogony, which is a highly contested and probably artifactual mode of reproduction. Doesn't reproduce that way, but it's an observation that's probably an artifact of culture; and then endodiogeny, which is the internal binary fission that occurs inside the cell. So what Feng Jin did was just to do high-resolution microscopy. So he had a very good microscope, and he labeled the nucleus with DAPI, it's a nuclear stain, and the mitochondria with mitotracker red. So it's a mitochondrial stain. So if you look at a typical cell, okay, you can see this, so you're zooming into this region, you can see here the nucleus is starting to divide, it's pulling apart, but just neighboring the nucleus, you see mitochondria. In some cells, you see the mitochondria further apart, and it has its own the nucleus pulling apart, and the mitochondria also associated with it. Many cells have nucleus at the opposite poles of the parasite, and here you see this. So this cell has polar organized nuclei with mitochondria. And then this is something very typical. You see what you call cytokinesis. So like in typical mitosis, there's always a furrow that splits the cell into 2. Okay, so this is typical. Nothing surprising there. But we also see parasites with 3 nuclei, 4 nuclei, and even 6 nuclei. Right? So this is what all of us see. Now, I'm not sure if anyone has done this, but what Feng Jin did was to measure cell number as a function of cell size. So whenever he counts one nucleus, he'll measure the diameter of the cell. And what he noticed is that cells with more nuclei are larger. Okay, so you have 1 nucleus, 2, 3, 4, or 6. The 6 nucleated cells are typically larger. So it hints at something now. Maybe you have some idea of why we start to see cells of such varying nuclear numbers. So we think there are 2 ways in which you can generate these multinuclear cells. One way is in which you have a cell with 4 nuclei, or we call this 4N cells, but one of the nucleus starts to divide ahead of the rest, right? So this one starts to divide, and then you have this cytokinesis furrow, the cell breaks into 2, then you have a 3-2 appearance. In this particular cell, you see this happening. There are 3 nuclei. The 4th nuclei has already started to divide. Okay, so there's a bit of a loss of regulation in nuclear division. Okay, now here you can see 4 nucleus, then you have asymmetrical division giving a 1-3 kind of outcome. So you see in this particular cell, plasmatomy-like replication, a 1 nucleus, cytoplasmic, cytokinetic furrow, and then 2 nuclei, so or rather 3 nuclei, right? So you get a 3-1 appearance. So this is interesting, but let's take a step back and ask the question, how do mammalian cells divide? How do cells divide in general, especially eukaryotic cells? So in eukaryotic cells, there are these filaments called microtubules. They are very important for cell division, okay? And in this cell, a resting mammalian cell at interphase, you have a centrosome. It's where all the fibers come out from, right? And these will start to arrange themselves. This centrosome will divide. It would then go to opposing poles of the cell and form the mitotic spindle. The mitotic spindle helps separate the chromosomes and nuclear material, right? And then it goes through different distinct phases. And if you remember your cell biology, it ends in telophase, and almost immediately after telophase, there will be a division. This is to ensure that equal parts of the cell is split into the daughter progeny. You want equal numbers of mitochondria and nuclear content so that you maintain fidelity of the cells. Okay. Now, one thing that's perhaps not often talked about are these little microtubules on the sides. These are called astral microtubules because they look like stars when you label them. Okay, so these are astral microtubules. They're very important for positioning the cell during division. So when Feng-Chen labeled microtubules of blasto, it was interesting. You see these very pretty star-shaped microtubules coming out from the centriole, right? And he sees this in all the cells, okay? So he sees this star-shaped emanations coming out from the nuclear region, and they look very much like the astral microtubules. What is unusual is that they're always there. In dividing cells, they're only there during division, okay? So the parasites exhibit these astral microtubules from the nucleus, and typical of mammalian cells at interphase. What happens when you have a dividing cell? Do you see the mitotic spindle? We don't see the mitotic spindle. Okay, so in this particular cell, you can see the astral microtubules, you can see the centrioles, but you don't see very distinct mitotic spindles. Okay, you still see the astral microtubules, even though these are 3 nucleated cells. So the newly divided nuclei lack typical spindle assemblies seen in mammalian cells. Now, is this reality or is this something that we miss? Actually, I'm not sure. My opinion is that it could have happened so quickly that we missed it, because I cannot imagine a cell dividing without mitotic spindles. They are so important and conserved for division. But it's not obvious in these specimens. So then what determines this cleavage furrow? I mentioned in mammalian cells, after telophase, the cell will split into 2. In Blastocystis, this doesn't happen. And one possibility is the fact that the nucleus needs to travel all the way to the other end of the cell before division can occur. Okay? So when the nucleus divides, it has to take time to make its way to the other end of the cell, and then division occurs. One suspicion we have is that these astral microtubules actually determine the cleavage site, and not so much the mitotic spindles. Okay, so we see this 2 cells with very typical binucleate cells, polar nucleus, and then you can make out the astral microtubules. So do these microtubules determine the cleavage site? When we see a tetranucleated cell, again, you see these astral microtubules, right? And if we are correct, then the cleavage site will occur here at the region where there's least or lowest density of the microtubules, right? We don't have the answer right now. This is preliminary data. I just wanted to show you something quite interesting, something compelling, but could also explain why we see such heterogeneity in cultures. So the terminal step in cell division is cytokinesis. So the cell undergoes a division, constriction between the two daughter cells, and it splits into two cells. In mammalian cells, you have a conserved structure called the ectomycin ring. Okay, it's made of actin, myosin, and then kinases. Okay, that because it's an energy-requiring process, it requires ATP, it requires phosphorylation, and so the ectomycin ring can contract and split the cell into two. In yeast, it's a different set of proteins. It's the checkpoint proteins that helps in the budding. In bacteria, it's a tubulin homolog called FTSZ, or the Z ring, that helps to split the cell. If you look at the genome of Blastocystis subtype 7, we use subtype 7 as a reference, it has most of the homologs of the ectomycin ring. It has actin, myosin, and Rho A. It doesn't have RAC1. Doesn't have the yeast homologs. It has the bacterial homolog, but tubulin is very common, so it's not surprising that we have a tubulin hit with bacteria. So do we see this concentration of actin filaments in dividing cells? So when Feng-Jin looked at non-dividing cells, or cells with one nucleus, you find that the actin is spread relatively evenly throughout the parasite. But in cells that have polar nuclei, which means that they should be dividing at some point, you see a nice equatorial localization of actin. And you see it from two cells here. So the actin filaments do accumulate at cell equator and likely initiate ectomycin constriction events. Okay, so this is a working proposal of how we get all this bewildering array of nucleotypes. We propose that it's these astral microtubules that determine the cleavage site. And this is a typical cell cycle that we would see most of the time. 1 to 2, and they split to 2 daughter cells. We do see 4N cells. 4N cells can split this way or this way, right? Okay, if they split either way, you get binucleate cells, which then divide to your single nuclear cells. These cells can split asymmetrically to form a 3N, 1N, and the 3N cells can divide one more time to result in your 6N cells. Okay, and these 3N cells can go on to become 2N and 1N. So based on what we've observed, we propose that this is happening, okay, but this is something of a work in progress, something that still has to be further defined. But for the first time, we are able to label the Blastocystis microtubule complex, and it does suggest that nuclear division is loosely coupled to cell cycle checkpoints and cell division. So the nucleus divides, but it doesn't necessarily have to. The cell doesn't necessarily have to divide after that. Okay, this results in your multinucleate cells in culture, and therefore the loose control of nuclear division and cytokinesis could explain the heterogeneity of Blastocystis morphologies. What we want to do is to develop a live imaging system to observe cell division in real time. I think that's important to validate what we are proposing. We want to search for checkpoint genes or lack thereof. We think there wouldn't be many checkpoint genes because there doesn't seem to be much checkpoints in Blastocystis. And perhaps the use of certain inhibitors to study the effect on cell division. Okay. I guess we ask the question, what is the survival or what is the function of having such a heterogeneous population? And my guess is that it could be a survival advantage to have cells of differing properties. I think that could be something that Blastocystis prefers because it helps it to survive under varying conditions. So that's the first story, very preliminary, but I thought it'd be quite interesting to initiate discussions after the presentation. I'd like to shift gears right now and talk about Blastocystis in the context of the host. And we know that Blastocystis resides in the gut. And in the gut, you have many immune cells that would, at some point, interact with Blastocystis. In my lab, we have looked at many innate immune responses. We've looked at antimicrobial peptides. We've looked at inflammatory cytokines and so on. we thought that it would be interesting to look at regulatory T cells. These cells are very important for maintaining tolerance in the gut. And this work is driven by a postdoc, Lucas Wojcik, and Professor Nick Gascoigne, who's a T cell expert. So why regulatory T cells? We call them Tregs as well. And these cells have an important role in regulating and suppressing other cells in the immune system. Okay? So Tregs control the immune response to self and foreign particles, like your microbiota antigens, and help prevent autoimmune disease. So the reason we are not raising an immune response to our current microbiota now is because we have many Tregs in our mucosa that prevent inflammation. So they are an important layer of intestinal defense, and they suppress immune responses against innocuous food and your microbiota-derived antigens. They do secrete an anti-inflammatory cytokine called IL-10, and any impairment of Treg function predisposes tissues to inflammation. All right? So here you see in a healthy gut, you have intact microbiota, a healthy population of Tregs that suppresses inflammation, and then you have the counterpart, Th17, which is inflammatory. So you need a nice balance between the two. If there's a disturbance, maybe through dysbiosis or other mechanisms, your Treg population goes down, Th17 goes up. This puts you in an inflammatory state. So the question we ask, what effects do blastocystis antigens have on Treg function? Okay, and what Lucas did was to expose T cells to either isolate subtype 7b or subtype 7h antigens. Okay? And then he tried to develop or differentiate these T cells into Tregs. So these are CD4 cells, and then if you add the right ingredients, you can make them Tregs. So how does blastocystis affect the development of Tregs? So in the control cell, over time, he manages to get about 50% Tregs, okay, in the absence of blastocystis filtrates. But in the presence of isolate B, only 35% of the CD4 cells manage to become Tregs, and isolate H has less effect on Treg differentiation. Okay, so if you look at just this population here, you see that the percentage of inducible Tregs are quite high in the control and lower in the CD4 cells exposed to blastocystis. So the filtrate derived from the culture of blastocystis isolate B had a substantial, significant impact on in vitro generation of Tregs, which was reflected in a decreased number of converted regulatory T cells. So blastocystis hampers the development of Tregs. Now, if you took those Tregs that were inhibited, we ask a second question: how well do they divide? Once you make the Treg, they should divide. Okay? And we study division by labeling the cells with a fluorescent marker. Every time the Tregs divide, the fluorescence will halve. Okay? So in the control cell, when you first label it, the population at 1 would be high. After one round of division, you have a high population in 2. That means these are lower fluorescing cells, these are higher fluorescing cells. After some time, the population will shift to the left because they get less and less bright. So here in control, the cells have already gone through one round of division, so you have a halving of the fluorescence. In the blastocystis exposed population of Tregs, they still mainly remain in the first step. They don't really divide. Okay, isolate H looks very similar to the control. Okay, so again, if you take the different groups 1, 2, and 3, and you measure the level of fluorescence in these levels, you find that in section 1 or the marker 1, that's where the cells have just started division, you find that most of them belong to isolate B exposed cells. Okay, and the rest have gone on to divide. So there's impaired proliferation of those iTregs that were exposed to the filtrate derived from isolate B. Okay. So the next question we ask, we go a bit deeper. These iTregs, in the absence of blastocystis, once they've been exposed, we know they are a bit impaired, but how do they function when they have to influence another cell? What they have to do, the Tregs have to suppress CD4 cells. Okay? How do they suppress CD4 cells? They prevent them from dividing. Okay? So what Lucas managed to do is isolate roughly equal populations of Tregs and CD4 cells and ask the question, what is the effect of these on these? Okay? And they have a suppressive effect. They should prevent these cells from dividing. That's how they prevent the immune response from flaring up. Isolate B, exposed Tregs, did not stop the CD4 cells from dividing. So the CD4 cells continue to divide, showing that the iTregs that have been exposed to blastocystis do not function. Okay? So what this suggests is the iTregs generated in the presence of blastocystis isolate B, supernatant, was characterized by decreased suppression activity, unable to stop the division of CD4 cells, suggesting that these compounds produced by blastocystis leads to a stable reprogramming of the iTregs. These iTregs are stably unable to affect the CD4 cells. I know it's all very confusing. I'm also confused because it's immunology. Okay? So I want to summarize it over here. So we have shown that blastocystis soluble factors are able to inhibit Treg development. So that's the first thing. These Tregs that do develop exhibit impaired proliferation, right? And are unable to suppress the proliferation of CD4 cells, cells important for the immune response. So it suggests that certain isolates of blastocystis are able to impair multiple Treg functions, okay, from development to division to affecting other cells. And this could lead to an inflammatory state. What are our future directions? We want to assay for blastocystis induction of the counterpart, the Th17 cells, which are often working in partnership with Tregs. And we also want to analyze these populations in the acute mouse model that we have developed and published recently. So that's the second study. And it's come to a point where we can make some conclusions, but we are looking more at the mouse model to validate our observations. And the last study I'd like to talk about is the most mature study that we have here, and it's done in collaboration with Cynthia's group. This is Feng-Chin, our postdoc, with Melissa and my other postdoc, John Yason. And this has to do with an enzyme that is found in a lot of bacteria, a lot of gram-negative bacteria. It's called tryptophanase. Here is the 3D structure of an E. coli tryptophanase enzyme. And this is the blastocystis 1. It is found mainly in prokaryotes. What is it doing in blastocystis? It was acquired horizontally, probably from a bacterial ancestor. Okay, so if you look at these two structures, I can't even tell them apart. They are so closely related. This project has an interesting origin. About a year ago, I received an email from Andrew Roger. I've never met Andrew, but he sent me this preprint of a very nice paper where he mentions he sequenced subtype 1, NAN2 strain. And the paper, the highlight of the paper is that 2.5% of the parasite's genome was acquired horizontally. Doesn't belong to blastocystis. It came from somewhere else, either mammalian host or from bacterial bacteria. Okay, so this lateral genes transfer was proposed to be important for adaptation to the hostile gut environment. So I read the paper, and what caught my eye was just one sentence among, it was a very lengthy and impressive paper, but in the paper it mentioned that blastocystis had many pathways involved in nitrogen metabolism, and one of the horizontally acquired enzymes is tryptophanase. Okay, tryptophanase catalyzes a very important reaction. It makes indole from tryptophan. The amino acid tryptophan is converted to indole through tryptophanase. Okay? Now, Blastocystis is not the only parasite that harbors this. I think Trichomonas and Entamoeba also have tryptophanase. Okay? But there's a very simple test that one can do to confirm whether Blastocystis has tryptophanase, and I will show you the data later. Okay, so if you were to just do a blast on E. coli tryptophanase against the Blastocystis database, you find very nice hits. But for the various Blastocystis orthologs, they all cluster together, quite closely related to Entamoeba tryptophanase. Okay, and they do cluster together with the bacterial counterparts. So there's a highly conserved tryptophanase or TNAA ortholog. There's a test that you can do for those who are microbiologists among us. When you want to detect E. coli, there's an indole test, and you add Covax reagent. And when there's indole, the Covax reagent will turn dark red. Okay? Those organisms that do not produce tryptophanase, you will not see any color reaction, as is the case with your medium and your tryptophan control. I got really excited. This test is very easy to do, so I asked John Yason, my postdoc, to try it out. And he tried it out on 3 subtypes, 1, 4, and 7. They all produce indole, large amounts of indole. Okay? So Blastocystis 1, 4, and 7 produce indole. I'm sure the other subtypes do as well. It's yet to be tested. If you wait for 3 days, the amount of indole is 1 millimolar. That's very high levels of indole in culture. So we are very confident that Blastocystis produces indole. Feng Chin then cloned the gene, expressed it, and did some enzyme studies. All right? So he studied the kinetics of the enzyme as it sort of catalyzes the formation of indole from tryptophan. So here, as you add more tryptophan, you get more indole, right? Now, it took some revision for me, but you remember there are things like Vmax and Km in your biochemistry lectures. I had to dig my memory as well. But Km value is important. Now, the Km value of Blastocystis tryptophanase is 10 times higher than E. coli. That means it has 10 times lower affinity for the substrate. Higher Km means low affinity. Okay? So this is really demoralizing. It's worse than E. coli by 10 times. Okay? But other features were quite typical. If you mutate the active sites, the catalytic activity is abolished. There's a temperature dependence and a pH dependence, typical of mammalian enzymes. Okay? But at this point, the project stopped because my postdoc said, it's lousy, it's worse than E. coli, 10 times worse. What should we do? So I reasoned with him. I said, Feng Chin, why would it need to make indole when there's so much E. coli making indole in the gut? There's already about 1 millimolar indole in the gut. Why make some more, right? So I said, why don't you test the other direction? Take indole, add pyruvate, and measure the kinetics when it makes tryptophan. You know that enzymes work 2 ways, right? And lo and behold, the Km in the opposite direction was extremely low, okay? 0.0048. This tells you that it has extremely high affinity for indole. So it rather makes tryptophan from indole than make indole from tryptophan. It is catalyzing the opposite direction. Okay, so he got excited again. I needed to keep motivating him, right? So, okay, maybe there's a story there. What is the story? Okay, we know that Blastocystis resides in the large bowel. Okay, in the large bowel, or we call it the big intestine or the colon, there's water metabolism, absorption of ions, vitamin K, and production of short-chain fatty acids. From the literature, we also managed to find out that the amount of tryptophan is quite low in the gut, but the amount of indole is quite high. okay, about 0.1 to 1. That's the range, okay? So the idea is that Blastocystis may not like indole. Okay, in that high concentration of indole environment, it prefers to make tryptophan. Okay, so this is the model we propose. E. coli and its counterparts convert tryptophan to indole. Indole is very quickly converted to tryptophan, and we propose that tryptophan is used by the parasite for growth. It would be interesting if indole was not good for the parasite, giving it a rationale for conversion to tryptophan. Okay? So does Blastocystis utilize microbiota indole for conversion to tryptophan? We think it does because at physiological concentrations of indole of about 0.1 to 1, Blastocystis doesn't like it. It doesn't grow in physiological concentrations of indole. But if you supplement tryptophan in the medium, it grows. So Blastocystis prefers tryptophan to indole. So physiological concentrations of indole and tryptophan exhibit negative and positive effects on Blastocystis growth, respectively. Right? So in order to prove this further, we decided to generate a tryptophanase overexpressor. Okay, so if E. coli can overexpress tryptophanase, making more indole, how would that impact Blastocystis? So in collaboration with a colleague, and we got his PhD student, Melissa, to generate an overexpressor. So this is to show you that she put in the tryptophanase gene was driven by a strong promoter, arabinose promoter, okay? So in this model, right, the E. coli now has an overexpressing tryptophanase gene producing more indole, which is readily taken up by the parasite to make tryptophanase. When you put these two together, right, it grows better than just wild-type E. coli. So E. coli that has overexpressing tryptophanase tend to help blasto to grow better. Okay, so this is if you take this data and you put it here, you can see that Blastocystis counts with E. coli, you normalize it to 100, you get about a 50% increase when you co-incubate Blastocystis with the overexpressing mutant. So the E. coli overexpressing tryptophanase enhance the growth of Blastocystis during co-culture. Right? So Blastocystis has an active tryptophanase, which is more efficient in converting indole to tryptophan than converting tryptophan to indole. The Km of Blastocystis tryptophanase is 10-fold higher than E. coli tryptophanase, meaning that it has a low affinity for the substrate. Low concentrations of indole promotes, but high doses inhibits Blastocystis proliferation. And so we believe that the horizontally acquired tryptophanase may have evolved to detoxify its microenvironment from indole-producing tryptophan. So we know that Blastocystis converts indole to tryptophan, okay, and we know that indole has a role in the microbiome. It's a signaling molecule. It has many roles in the immune system as well. If there's an overproliferation of Blastocystis, how does that perturb indole metabolism in the gut? So these are our future directions. You want to study the role of indole in the microbiome. But something a little bit provocative here, Blastocystis produces tryptophan. Tryptophan is an important precursor of serotonin. Right? Serotonin is a neurotransmitter implicated in IBS. Okay? So the question is, if you have a high infectious dose of Blastocystis and you have a lot of tryptophan, how does that perturb serotonin metabolism? Okay, and this is another active area that one should consider. Okay? So I'm not going to talk about this. I'm going just one slide to tell you that we have a very exciting project which my postdoc, John Yason, will elaborate on tomorrow. It has to do with Blastocystis, members of the microbiota, the gut, and an animal model. Okay, so just to keep you excited, don't go home after this. Come back tomorrow, please, and attend this talk. It's a nice collaboration between Professor Chang Yong Liang, who's an immunologist and animal infection expert, Chin-Wen, Yiran, and John. Okay. I have only one picture on Singapore. right? It is a part of Singapore that is very lively. Last night I went out for drinks with some of Juan David's students. Really loved it. And if you love that, you will love this. This is a part of Singapore that's full of pubs. And it's a small country, but we offer a lot in terms of research and culture. So thank you for your time.
Yes, so detailed and so excellent works. Thank you so much, Kevin. All of them are very new, and we learn, and we have also many questions about that.
Yes, please ask.
And you have three different
projects,
projects.
Yeah.
And I think we can start first one.
Okay.
It is about the blastocystis and cell cycle, especially. It is so interesting study. So I think you have many different laboratory equipments to make all this study. So good. And it is so nice to learn why some blastocystis have only one and other have one more nuclei. So it's an interesting study. And any question for this project?
Juan David?
Yes.
Thank you very much, Kevin. Interesting talk. I have one question regarding the cell cycle. So you showed that there are some stages where blasto can be tetraploid, right?
Yes, that's right.
Have you found any association between that tetraploid stage and the morphotype?
So what we know is that the higher the ploidy in general, at least in the subtype that we observe, the higher the ploidy, the larger the cell, right? So if you have 1, 2, 4 nucleus, they tend to be over 20 microns in diameter. And what that hints to me is that when the cell divides, the cytoplasm continues to accumulate and doesn't divide. So the cell division is very poorly linked to nuclear division. So the nucleus can divide and the cell doesn't divide. It goes through another round of division and the cell still doesn't divide. And so we have this giant cells with multiple nuclei. Now, have you seen giant cells with one nucleus? I think they're hard to find. I think these will be quite aberrant. Normally, your big cells are multinucleated, and I think that's because cytokinesis has not been activated.
Okay, and do you know if somebody has sequenced, I don't know, maybe Andrew knows, the tetraploid stage? I mean, does the genome change if obviously the genome length will change, but in terms of heterozygosity, do you think that might change or not? I mean,
so you're hinting that it could be an opportunity for gene exchange and recombination.
Exactly.
That's very provocative, and I think Andrew might have some inputs on that. Is there a way we can test? Hisao showed that he can take out individual cells using micropipettes. Is it possible to do single cell analyses of these giant cells and look for recombination events that occur more than mononucleated cells?
I'm a little confused about the use of the word ploidy here, because what you're describing is multinucleate states, but the ploidy of each nucleus
remains the same. Right?
Yeah, do we know what that is? I mean, we think it might be diploid based on genomes the genome that we did, but I didn't get a strong feeling about that. So do you have a sense of that?
I wouldn't know about the ploidy, so I think we need to go one step further and do organelle isolation. So if there's a way to isolate the individual nucleus and then analyze the DNA content there, or there may be some methods to just pinpoint at these cells and measure the relative fluorescence or do some fish analysis as well. So at this stage, it's an open question. We don't really know the ploidy of these cells. Yeah. Yes.
Anything?
I used to work a lot with cultures, and it's something very rewarding, actually. And those of you who haven't worked with cultures, I suggest you do, because it's actually quite easy. And actually, when you've subcultured your culture for some time, you'll see that you get rid of a lot of the debris, the fecal debris, and you have almost like a pure suspension of cells. And it's very nice to just look at that. And you have them in all sorts of forms, so please go ahead and look at that. My impression is that when you have really fresh cells and have a regular subculturing, you have very tiny cells. But if you leave a culture unattended, you'll see these bigger and bigger giant cells. Is that also your experience?
So I think that's an observation that many of us share. Those who look down microscopes all day, you tend to notice, and it was raised at the workshop as well. And again, it may support our observations, because if the conditions are right, then cytokinesis can occur as it should. But if you stress out the parasite, it loses its timing and regulation, and then it starts to form these giant cells. So I think the giant cells could be a surrogate for how stressed out the cultures are. Now, whether that's beneficial to the parasite, is it a healthy response, or is it, do these giant cells continue to die? We don't know. I think that's another open question. Therefore, this life culture systems, life imaging systems for imaging the whole cell cycle of blastocystis, I think it's important to answer these questions.
So we cannot define the number of nucleus in blastocystis when they are stressed. We cannot define the exact number of nuclei when blastocystis are stressed or where cytokinesis is not performed.
Yeah, so we can label the nucleus with Herx or DARPy, and we can count them, and that's why in these few large cells, they have 6 nuclei, and they are about, I think, 30 to 40 microns in size. That's actually quite large. So it's possible. Yeah. Was there a question behind? Yeah, Andrew?
Yeah, that was an amazing set of stories. I was going to ask about the indole story, and am I right? I think tryptophanase breaks down tryptophan into indole, but ammonia as well. Is that right?
I think ammonia is a byproduct, and then there's pyruvate as well, I think.
Yeah, so I think ammonia is coming off there.
Ammonia, yeah.
Yeah. So then I wondered if the other metabolites that are produced by it, have you checked their effect on growth? Like.
All right. Okay, I see what you mean. That's the reaction here. 1 second. So in this direction, tryptophan, yes, there's ammonia, pyruvate, and indole. So yeah, we have not checked. Actually, that's a good question. We have not checked whether it's actually a bystander effect. Something else could be impacting.
Because if you raise the amount of indole, but don't raise the amount of pyruvate and ammonia, then it can't do this reaction, and pyruvate and ammonia might become limiting or something.
Right. Yeah, so I think we have to control for the other substrates as well. Thanks.
If any question, first talk. Any question? And you?
Sure.
Yes.
Yeah, we're flexible here. No.
Yeah. Okay.
No.
Hi. The data you showed about the Tregs, that was all in vitro, right?
Yes.
But you guys do animal model work too, so I was just
Yep.
wondering if you had any plans or were currently looking at how Tregs are responding.
Yeah, we're doing that
in
right
vivo.
now. Yeah, but I think it's being done while I'm here. So Lucas is supposed to infect the mice and then extract the tissues to see whether there's a destabilization of Tregs and maybe an upregulation of Th17 in the tissues. Yeah, so I think we need to complete the story with the mouse model. So that's lacking right now.
And you're just going to look in the guts or in like peripheral immune tissues as well?
I think they'll just take everything. They'll take the blood, they'll check everything. But I think the focal observations would be more relevant. Yeah.
So you were explaining in the workshop that the ST7 was the one that was more virulent than the other subtypes in your lab.
Yes.
So I was wondering, the data on the Tregs was all with ST7?
Was with ST7.
Have you
yes.
tried it with the other subtypes?
That's a very good question. And we have to, in fact, John, we're going to, right? Did Lucas ask for subtype 1? Yes, I gave him the subtype 1. Okay, great. I'm glad you answered that. So, yes, you're absolutely right. Because if we want to show that a particular subtype is virulent, then we have to show that the A virulent subtype doesn't exert such an effect. So the reason that we didn't do it initially is, and Lucas explained this to me, it's extremely difficult to do these Treg experiments. It takes 5 days to differentiate them, another 5 days to do for other experiments. So he wanted to be sure there was a phenotype before expanding to the other subtypes. But thank you, that's a very good point.
Kevin, can I ask
Sure.
you, which subtype, do you have some axenic strains? Which one of them? All of subtypes?
Okay, so I can tell the community that I have many isolates. Subtype 1, subtype 1, 4, 7, and multiple kind of genotypes or multiple isolates of each subtype. Am I missing any? 147, right, John? So 147. We also have reptilian isolates, if you're interested, from iguana, from snake, from tortoise, from cockroach. My former supervisor was positive. So we also have his isolate. So it's a special, very special isolate.
Okay.
So if you need these isolates, please email me, and I will send you either the DNA or the live cultures. Okay, just contact me.
Yes, we have a time. More question? Yes, Andre?
So you didn't make too big a deal of it, but it really did look like the isolate B and the isolate H behave differently, and they're both subtype 7.
Yes.
And that looked quite striking.
Yes. I'm glad you noticed that.
Yeah.
So they are from subtype 7, and they have very differing phenotypes. In fact, we have 5 isolates of subtype 7, and they all behave differently. In terms of drug resistance, subtype 7B is metronidazole resistant, H is sensitive. H damages tissue cultures to a large extent, B doesn't do it as much. So there's a lot.
But in this case, B was more inhibitory.
So in this case, B is looking more like a pathogen than H. So we thought about that, and we think that H damages the cells by different mechanisms. It's a different virulence mechanism. It's certainly not through immune modulation. Again, the mouse model will be important to verify this.
Wow. Thank you.
Yes, maybe last question. We are looking forward to your papers about these projects.
Thank you. I will push my postdocs to publish. Thank you very much. I'd like to thank the previous speaker for giving his talk, and now we'll start the oral session. So in this session, we will focus on the topic of molecular and cell biology in the context of blastocystis. And our first speaker in this session is Joel Martinez Ocaña. Am I correct? And he will be talking on the identification and immunolocalization of an alpha-L-Fucosidase in blastocystis.
Good afternoon. I am Joel Martinez. I want to thank the organizers of the Congress for accepting my work.
Joel, please use the mic. Use the mic.
Okay.
Yeah, louder. Thank you.
I want to start to taking directly about this paper in the complete genome of the Blastocystis 7 subtype wide sequence. This paper proposed the scheme with different secret proteins and factor virulence identified. Among the different secret proteins was Alfa-L-Fucosidase, an enzyme this part of the glycoside hydrolyse family. The ALF is an enzyme and glycosidase that catalyzes the release of terminal fucose of mucin found in the intestinal epithelium. In bacterials, such as Helicobacter pylori and Bifidobacterium, it has been observed that fucose produced by the action of ALF in the intestinal mucin are used of the source of carbon for metabolic pathways, these bacterials, which allow the tainment coincide the intestine. In Helicobacter pylori, an association between increased expression of AL and pathogenic strain has been observed. It's important to mention that the literature, there are no reports of ALF identified in parasite. And for this reason, we're interested to identify in blastocysts. Objective, identify of ALF from blastocysts, establish the in vitro culture of blastocysts, identify alpha Western blot and blastocyst protein extract, immunologize ALF in blastocysts. These methods results. This protein, I have the fucosidase domain, in the image below is the correspond to ALF Bifidobacterium, time the signal peptide, domain fucal Alfa-Fucosidase, and immunoglobulin type domain. These images of the culture, more images. Also, the blastocystis is stained with chalcofluoride in fluorescent microscopy. Also, analyze culture of patient or carrier blastocysts to find the different times generation times in between two cell types. In electrophoresis in one dye, the show is the protein 256 kilodalton is the molecular weight expected for the ALF. and the Western blot identified the same proteins, 56 kilodaltons, and today is the same protein recognized in the same antibody. Immunofluorescence, the same antibody are recognized blastocyte isolate from the real phase. It is clearly recognized. Blastocyte isolate from no real phases, reaction negative. Then alanine, alanine where I may finding large regions of identities in their different subtypes of blastocysts. In three observe three clades and one ALF subtype 4 and 8 and 3, another subtype 7, 6, 9, another is subtype 1 and 2. All the realized prediction structure of proteins of different subtypes of blastocysts, 1, 2, 3, 4, 6, and the results show identities, only small difference in the amino acids structure, Alfa-L-helix or beta strand. Conclusion. Crude protein extract from two strains ATCC of blastocysts and prepare. It was possible to identify ALF white Western blot in blastocyte protein extract. Is why identified by immunofluorescence ALF in blastocyte, for example, with diarrhea. In the electrophoresis 2D of strain of blastocytes, though two strong expressive proteins were identified, which also recognized of the anti-ALF antibody. In the prediction of the protein structure of the different subtypes, great similarities were found, only with small difference in the number of sites of the structure alpha helices or beta strand were observed. Finally, 2/10 of team of laboratory. Here, Dr. Pablo Maravilla, and the National Council for the Science of Technology, CONACYT in Spanish, for the support of this project. Thank you for attention. Thank you for my English.
Thank you very much, Joel. Thank you for, and I really applaud you for working on this very difficult project. I think while we acknowledge that blastocystis is very easy to culture, biochemistry and molecular biology of the parasite is not easy. So a good job. Thank you for all your effort to do this. And I'd like to open the floor to questions. Any questions from the floor? So maybe while waiting, I could ask you a couple of questions. So you mentioned that these are ATCC isolates.
Yes.
They are axonized or pure isolates, or did they come with some bacteria. Are they pure? Pure isolates.
Two
And
strains.
two strains.
Two strains.
One from a diarrheal
Separate.
patient and one from a healthy patient.
With faces diarrheal and no diarrheal is the same subtype. Interesting.
Same subtype. What subtype is it?
One.
Subtype 1.
Subtype 1.
Okay.
I try axenic cultures. It's very difficult. It's very complicated. I tried.
Okay. So your labeling of the Fucosidase, was it a surface labeling or was the Fucosidase inside the parasite?
I don't know, really in granulates or periphery.
Okay.
I don't know.
But it's a bacterial enzyme, isn't it? Fucosidase, is it a prokaryotic enzyme or?
Yes.
It is, right?
Yes, in bacterial.
Okay.
Yes.
But in bacteria, is it on the cell surface or?
Two types of ALF.
Okay.
One is I can secrete, and the other enclosed
intracellular
membrane.
Or membrane encoded.
Encouraged.
Okay. So one suggestion is that Blastocystis can be separated into fractions. So you can actually lyse the parasite by freeze thawing. Then you can spin, do a very hard spin. And you can then take the supernatant and the membrane fraction, do your Western blot again, and then you can determine whether it's a membrane encoded protein or whether it's a membrane or cytoplasmic protein.
Complete protein.
Just fractionate. No, the parasite itself. You fractionate the parasite, then you use the antibodies.
Fresh parasite.
Run the soluble
Yes.
fraction and the membrane fraction.
Yes.
Yeah, you can see me after. I can talk to you. Yeah. Any other questions from the floor? Yes, Rodolfo.
Can you see again the immunofluorescence of the your sample? La immunofluorescencia. Nuevamente. The next one, please. Yes, it's impossible. It's impossible to detect the localization of the really. So I would expect it was on the surface of the parasite, but it's not. It's very difficult.
Maybe the fixation and all that needs to be optimized a bit.
But not all the cells were marked.
I try now identify granules of type Areny. I call this protein. I hope.
So most of these from culture were bacular forms.
Vacuolar forms? Are they vacuolar forms? Yeah.
Okay.
Okay.
Okay. That's very interesting, eh?
Yeah. If no more questions, please help me to thank Joel for a very interesting talk. Thank you very much. So we have another interesting talk lined up. It's by Liao Chen-Chieh from the National Cheng Kung University, Taiwan. So we welcome him and thank him for making that long journey to Bogota. So glad to have you here. And he's working on a cysteine protease of a Blastocystis subtype 7. He's going to talk about how he characterized this particular protease. So, Chen-Chieh, over to you.
Thank you, Dr. Kevin. Then, my name is Liao. My name is Zhengjie Liao. I come from Taiwan. So today my topic is identification of cysteine protease of Blastocystis subtype 7. So in outline, there are two parts. The first is evaluation of lipid accumulation affect the pathogen effect of Blastocystis hominis. The second part is to to identify the cysteine protease of Blastocystis subtype 7. And in our study, we use the subtype 7. This subtype 7 is from the Dr. Kevin. So this is why I can come here to share my study. And when I culture the Blastocystis, we can see the two morphology, the vacuole form and the granular form. And we also see the nucleus and the mitochondria-like organelle. So in previous study, the data show that after co-incubation, the Blastocystis will induce the inflammation, just increase IOA of the HT29 cell. And in other part, this data is from Dr. Yoshikawa. He find the Blastocystis will accumulate the lipid in the central vacuole, but the function of which is now well known. So this is why I study the lipid accumulation of Blastocystis. And in recent year studies, they found that the Blastocystis will release the cysteine protease to increase the intracellular permeability, induce some digestibility passage like the IBS. And in this result is from Dr. Kevin. They found that after co-incubation, the Blastocystis will increase the COCO2 cell disruption just by disrupt the cell type junction. So in my study, we just follow the previous study. Here we use the HT29 cell. After co-incubation, we can see the Blastocystis induce some cell disruption, like this movie show. And then we want to know how do the gene regulate after Blastocystis infection. So here we use the microarray. From this microarray data is from the 163 human oncogene and the kinase. From these genes, we find the 10 genes will significant change after Blastocystis infection. There are 4 genes are significant upregulate, and the 6 gene will significant downregulate. So this is 10 genes. And we focus the CREP2. The CREP2 is most significant upregulation after the Blastocystis infection. So next, we want to confirm this microarray data. So we first, in gene expression level, we use the real-time PCR to analyze these 2 genes expression. First, in CREP2, we can see the results is time dependent from the 6 hours, 24 hours, and 48 hours. The CREP2 expression is upregulated. And in the PCNA expression, is also in time dependent. The PCNA is significant downregulate. So then we also confirm by the protein expression label. Here we use the Western blot. And we also see the same condition. The CREP2 protein expression label is upregulated by time dependent. So then we also we use the immunostaining for CREP2. We also see the CREP2 the expression will upregulate after the Blastocystis infection, and we also can see the immunity staining. We can see the CREP2 is expression in the nucleus. So we found from the microarray data, we find the CREP2 and PCNA, these 2 gene expression, and we want to know the protein-protein interaction network. So here we use the string list database and find the other proteins interaction. So here we base the string database, we find other genes like ISR alpha and ISR, RAR alpha and TP53 and the ILA. And we find this other 4 genes, and we can see after the Blastocystis infection, the IR alpha and the ILA have well significant upregulate. But the ISR alpha and the TP53 is not a significant change. So here we find this gene will involved in retinoic acid signaling pathway. So after the Blastocystis infection, will induce the HTT29, the CREP2 upregulate, then the CREP2 will bind with the retinoic acid, then it will change to locate the nuclei, and this retinoic acid will binding with the IR alpha, then we will change to the target gene, like the PCNA or ILA. And we can see from our result, the ILA expression is up and the PCNA is down. And they may be induce the cell goes to apoptosis or growth inhibition. Then here we use another cell line. We use the CACO2. And in this result, we can see the same situation. After the Blastocystis infection, will induce the CACO2 cell will disruption. And we try to study the lipid accumulation of the Blastocystis. So here we just construct a lipid-rich environment. So when we culture the Blastocystis, we add the extra lipid to create the lipid-rich environment. And in this lipid-rich environment, we can see the Blastocystis will increase its lipid accumulation. And after the Blastocystis infection, we can see the Blastocystis will induce the cell disruption. But in the lipid-rich environment, they can induce higher cell disruption area. So this is after quantify the data. So we can see in the lipid-rich environment, the Blastocystis induce the higher cell disruption. And we also analyze the ILA expression. We can see in lipid-rich environment, the ILA expression is higher than in a normal median. So here we also try to detect the CREP2. So in this lipid-rich environment, we also can detect the CREP2 expression is higher in the lipid-rich environment. And in this lipid-rich environment, we also found the 16 protease in his total activity, and the protein expression is also has the higher activity and expression. So here we try to identify the 16 protease of the Blastocystis ST-7. So in previous study, they show they have in Blastocystis they have 9 16 protease. In the high molecular weight, they have 5 16 protease, and in the low molecular weight, they have 4 16 protease. And in Blastocystis squeeze 16 protease, they have here we can see the result have two 16 protease. The one is the ligament, the peptidase C13, and the other one is caspase peptidase C1. And we try to find the other sitting post in the Blastocystis. So here we use the NGS. We also can define the 6020 CDS, and after re-annotation, we just find the 4000 CDS can be annotation. So in this result, we find the 3 C1 family 16 peptides. We just call the CPA, CPB, CPC. We can see this. These three peptides C1 family, they have the peptide C1 domain. And we confirm this data, we just design the PCR primer. So we can, from this data, we can see they really have expression in the Blastocystis subtype 7. And the other way we find, from this data, we find the CPA, it will expression in different subtypes, like in subtype 1, subtype 3, and the subtype 7. So we think the CPA maybe is important of the Blastocystis. So here we try to purification this CPA. So this is what I do here. But this part is primary data. We just ongoing to experiment this. So we just get the recombinant protein. But this recombinant protein, we just try to prefer for large number. So here in the future work, we will use this recombinant 16 protease to incubation with the host cell to see this recombinant protein's function. So in summary, from our data, our data show in a lipid-rich environment, the Blastocystis will induce the higher cell disruption and also upregulate CREP2 and IO expression. And in summary, we can see after the Blastocystis infection, that will induce the CREP2 expression just through by the eye signaling pathway. And our results suggest that the influence of Blastocystis subtype induce the higher inflammation and disruption of host cell in the lipid-rich environment. Also, the mechanism is not yet understood completely, but our findings suggest that the lipid droplets are key factor involved in the mechanism of the Blastocystis infection. And finally, we find the 3 C1 family 16 peptides of Blastocystis ST-7, but the function we will study in our future work. So thank you, everyone.
Thank you very much, Chen-Chieh. Very, very interesting talk, covering 3 different stories, so thank you for that. Any questions from the floor? Yes, one right at the back.
At the beginning of your presentation, you mentioned that you found out that vacuola was accumulating lipids, right? Yeah. My question is, you mentioned that in poor lipid environments, there is a lower apoptosis rate. Do you think that perhaps an equilibrium between these 2 metabolic pathways may confer blastocystis how to say a higher pathogenic effect?
You mean in lipid environment and they will affect the Blastocystis metabolism
yeah.
to increase the pathogen effect, right?
Yeah.
In this lipid-rich environment, we find the cysteine protease, the activity and the expression is higher than the normal condition. And we also study the fatty acid synthesis pathway of the Blastocystis. We also find the GPTH and GLO and the G6PD, they were upregulated in this pathway.
Andrew?
That was very nice. Do you have any idea how Blastocystis takes up the lipids? In what form? Does it take up the fatty acids, or?
From the data not shown here, but from my results, I just see in the fatty acid pathway in the lipid-rich environment, and we upregulate the GPTH is involved in the glycerol, and the G6PD is involved in the free fatty acid biosynthesis, and the glycerol bind with 3 free fatty acids that will synthesize for triacylglycerol. So I think they may be to synthesis for the triacylglycerol. And from the lipid stain, we just we can see the lipid drop, but we don't know the concentration.
What kind of lipid do you add to the culture?
We use the nutrition from the hospital. It's a lipid mix.
A mix.
Yeah.
Yeah. Okay.
Thank you. Any other questions? John?
So just some technical questions. So did you differentiate when you did the EC29 plus Blastocystis, did you differentiate the EC29? Did you induce it to differentiate? I mean, yeah.
You mean in
yeah,
in CACO2 cell?
Yeah, the EC29, or even the CACO2 cells with because CACO2, I think they take 21 days for it to have this spontaneous differentiation. Then you get this characterized tight junctions, and with EC29, you can use sodium butyrate to differentiate, and then you get a more expression of tight junction proteins.
Yeah.
Yeah. So.
But I didn't study the tight junction in my study.
Yeah. Okay. But it could have some, effect on the.
I just detailed like the Leo one.
Yeah, something like that. Okay. And then 2 more, just some short questions. Did you do it in the incubation? Did you do it in anaerobic condition?
Yeah.
Okay.
You mean the incubation is not so an incubation. It's in the CO2 incubator.
Okay, so that means like the experiment for 48 hours, the Blastocystis was is exposed in oxygen, right? Yeah. Okay. And then last is what is the density of the Blastocystis you use for the co-culture with the HU29? What is the density? Do you remember?
Density.
But yeah,
I just
okay.
I can tell you later.
Yeah, thank you.
Okay, I have one question, Chen-Chieh. I have one question here. So you did an essay where you showed that the Blastocystis with lipid-rich media had more cytopathic effect on your tissue cultures.
Yeah.
Did you have a lipid control? Just lipid only added only to the cells.
I see in lipid-rich environment, the CACO2 cell is not so schematic change.
So you had a control where you added just the lipid-rich media onto the tissue cultures.
Yeah.
You did, because I did. Did you show it?
I didn't show.
Okay,
Yeah,
okay.
sorry.
So that's kind of one of the questions I had. Okay. Okay. Thank you. Any other questions? Okay. If not, please help me to thank Chen-Chieh for a very interesting talk. Thank you very much.
you.
Okay, we move on to the last talk of this session before we have our coffee break. So we have William Arony from Hospital General Dr. Manuel Gea González, Mexico, and his talk is entitled Post-transcriptional regulation of mRNA in Blastocystis species, a proposed pathway. Okay, William, over to you.
I prefer Arony. Sorry, I have two names. It's just a thing in Mexico. First of all, I want to thank the scientific committee to allow me to show, to have this oral presentation. I'm very happy that for the first time in a lot of time, I have not to talk about basic things of blastocystis because all of you have much of them. So in comparison, but I choose a non-traditional presentation, and later you want to see why. In comparison with the previous sessions, my research is just in diapers, or that's the truth. So I'm gonna talk about a proposed pathway for the transcriptional regulation of mRNA, and that's something that I started to work just a few months ago. So, as you can remember, most of the things of the basic traits of the basic biology of blastocystis are still unknown. We know that it has a range of hosts that has a lot of morphological forms, but I'm a biologist, and I hate to think that the only important thing of blastocystis is its relevance as a true pathogen or not. So, for me, my question starts with the granular form of blastocystis. But but I wanna start with another thing. Well, in eukaryotic cells, there are another structures besides those that have membranes that makes or that have a lot of specialized functions, like an aggregate, some cytoplasmic foci that forms under certain conditions. These structures are termed or labeled RNA granules, and you may start thinking why I'm interested in the granular form. These granules have a lot of functions, has related with most of the eukaryotic cells, single cells or specialized cells like the neurons. But what functions make? The three most studied RNA granules are the P-body granules that aggregates with mRNA and mRNA binding proteins, and these are intended to degrade mRNA that are not gonna go to translation. But there are other granules like stress granules that are a direct response for the eukaryotic cell, intended to avoid various environmental insults like heat shock, nutrition deprivation, many other things, inclusive parasitic invasion. So they have been described on germ granule, like germ. Another kind of RNA granules are the germ granules. Described in gonads of blastocysts of zebrafish and C. elegans and other cells. So for me, this research started when I was a PhD working with another things, but many of the things that was going on in the lab was based on blastocystis cultures, and people in the lab made some observations. For example, during at least in our lab, during in vitro cultures, blastocystis populations on the tubes changed from bacular to granular on certain time, and the proportion of cells or the density of cells never changed in these experiments, but the bacular forms decreased and the granular forms increased. So we thought that this could be just a stage conversion, and even in some points, even when new medium was added or passed to new test tubes, blastocystis never recovered, never grew. We had a lot of troubles there in that time, and we didn't know why. So also observations in differences under microaerophilic and aerobic conditions on blastocystis cultures were made during routine cultures. So I was starting to think that this could be another thing, and for me, the first response to that hypothesis was the paper published by Durga et al. A couple of years ago. If you remember, they isolated various subtypes of blastocystis and induced apoptosis by exposition to metronidazole, one of the most prescribed antiparasitic drugs, and they found at least 4 populations for blastocystis, for granular blastocystis. And I textually cite the paper or the authors that they say that this suggests that granular formation during apoptosis is a self-regulatory mechanism to produce higher number of viable cells in response to treatment. So why to relate this response with the RNA granules? Because RNA granules or stress granules are intended to contend to many kind of stress. So this is a stress, or blastocystis was under stress, and granules were formed during this exposition. They reported that for example, when subtype 3 has more granules and that granules were higher, viable cells, even when they were apoptotic, the number of blastocystis were more than the other subtypes. So they were some kind of difference inherent to genetic variation in species. So we started to think that. And for me, that I just obtained my PhD just last year. I was contracted like an assistant researcher. Well, where to start? For me, it's a quest. And I wanted to go to available information. What available information? Information available for RNA granules from experimental reports in other eukaryotic organisms. We made a deep search of experimental reports about RNA granules, and we looked more than 80 papers that were experimental, in which 17 different species of eukaryotes from mammals, for example, Xenopus, or even zebrafish, Trypanosoma, were reported, and we found around 208 proteins related to mRNA granules. So we looked for the 3 most studied mRNA granules, and we looked at stress proteins just for stress granules were 49. You can see here the numbers for each granule, and the granules shared by 2 mRNA granules or the 3 mRNA granules that are considered like the backbone of these kind of structures. So after that, we wanted to know if Blastocystis might have these elements to form mRNA granules. So we looked. These are 2 screenshots of the NCBI system about the genomes already sequenced of Blastocystis. As you can remember, there are at least, if I don't, but 4 genomes with different level of assembly. They are already proteins annotated inclusive. So we started to look for these proteins from other eukaryotes to look for homologous in Blastocystis data. So we found this is a representative image of the proteins that we found already annotated in the system. So these are the homologs in Blastocystis. There are a lot of proteins like poly-A binding proteins, hexoribonuclease, RNA, dead box RNA helicase, argonauts, translation factors, and another kind of proteins. What is the important thing here? There are traditional markers for this kind of granules. For example, RCK, also called P54. It's a protein that if it's mutated or knocked down, granules can be formed and cell can form stress granules to contend to a specific stress. So we found around 50 proteins. In black, there are the proteins that we are going to start to work, especially P54, where we have just an antibodies and started to make some tests on granular forms. Well, we made phylogenetic studies of those proteins, around 55 proteins that have been implicated in structuring or localizing mRNA granules in other organisms, but we are still fishing distant homologs in Blastocystis. So it's every protein or every family protein has its own complication, its own analysis. And we are making just right now bioinformatic analysis. So 2 of the key points that we have reached is that, for example, dead box helicases homologous families in Blastocystis are well conserved in sequence and domain architecture. So for us, this makes us propose that Blastocystis have all the elements to construct or to form the RNA granules, so that this makes us suggest that some part of the granules in blastocystis might be RNA granules. Another key point is that the PAP homologous family in blastocystis contain the conserved PAP central catalytic domain, which is involved in the nucleotide transfer, and are intended to use them to bind certain mRNAs, to take them to the RNA granules, and to reach its function. This is what we think that that might be happening when the mRNA are exported to the cytoplasm. They join certain proteins, certain mRNA binding proteins, and can be taken to the P-body to be degraded, or to the stress granules. Some of the proteins are shared by these granules, so it's usually that when P-bodies are present, stress granules can form. So this is just like a thought. And right now, we have preliminary results in localizing P54, one of the core proteins in RNA granules, in blastocystis granules during heat shock with antibodies intended to zebrafish that were given to a friend, a collaborator. But you know what? We made it on Jones medium. And bacteria also have this protein, and it's also highly conserved. So we are waiting for specific species antibodies to P54 to confirm that these granules are truly RNA granules. We didn't want to rush with images or something, because we might be making wrong assumptions. And well, when I knew that the Blastocystis conference will be held, I decided to come because I need help. I came here for guidance, for more experienced researchers. So I hope not to be boring. I want to thank Dr. Pablo Maravilla, Dr. Joel Martinez, Dr. Mirza Romero, Fernando Martinez, Cecilia Sampedri, Ernesto Maldonado, and 2 students that joined this earlier research. Thank you.
Thank you very much, William, for a very interesting talk. Any questions from the floor? We have time for a few questions. Rodolfo.
And I want to make an answer for all you, because I don't know much about which is the electronic conformation of those granules. Does anybody knows? Are they surrounded by membranes, or are only aggregate proteinases and mRNA aggregates? So in normal mammalian cells, they are aggregates, not surrounded by nothing.
No, they have no membrane.
What do you think would happen? I ask myself, and I ask everybody if someone knows. Dr. Yoshikawa? No. Because I have many doubts. Funda, what do you think about this? Because there are many kind of granules when you see, I see them especially, not in cultures, I see in patients. In cultures, mainly we see the bicular forms that they keep during many, many times we pass and pass and pass. They, in my experience, they do not transform easily in granular. We mostly see granular forms in patients. And I ask myself, are these real granules? Are they vesicles surrounded by something, or what are they?
That's okay. So maybe I can just add some input. Maybe Arun can also suggest. So I think historically, if you look at the old papers for blastocystis, a lot of electron microscopy was done. And actually, the word granular form is a catch-all term for anything that appears granular in blastocystis. So there have been reports of lamellar structures like multimembranous structures within the central vacuole. Some people have proposed that there are progeny in the central vacuole. And there are also lipid droplets and all that. So I think it's a very heterogeneous group of cells. I don't think the granular form is a distinct form in the parasite. I think it's a response to stress, and it responds in a very unpredictable and heterogeneous way. I don't think that the granular form recovers. I don't think you get progeny from the granular form, for example. I think it's like a dead-end cell. Rune, maybe you'd like to... Yeah.
Draw on my experience. So again, if you have a culture which has been sitting there for more than a week or maybe 2 weeks without subculturing, my impression is that you will see those granular forms. So I always thought that it was some sort of like degrading stage. So, and on the contrary, if you are very, very good at subculturing, you'll see those very, very small vacuolar stages, and that's what you see. So that's just my sort of lab experience.
So, Funda?
I think it is end stage of before dying of the cell. When I incubate for a long time without so much subculturing, and I saw like this granular formation. And the log time of the culturing process, the population of mostly vacuolar, and then you incubate, for example, nearly more than 5 days, you can change the population change to granular formation.
Thanks, Funda. So I think maybe an idea for you is to work on these aging cultures, because your granules are stress granules, isn't it? They're like stress granules, but you have RNA deposited in them. So maybe if you want to enrich for these granular forms, you could use older cultures, for example, and perhaps use axenic cultures. I think ATCC also, you can get the NAN2 strain, which is axenic. Yeah, you can try that. Okay. Any other questions? If not, thank you very much, William, for a very interesting talk. Thank you all speakers for keeping to time. Thank you to the floor for your very engaging questions and for your interest in these topics. So I'd like to call this session to a close.
Durán. She is a research microbiologist of Environmental, Microbial and Food Safety Lab of U.S. Department of Agriculture. A little bit short to introduce her background. Her current research is wide range from to develop molecular methods to detect zoonotic protozoan parasites and apply these methods to specimens obtained from farm, wildlife, and surface water for epidemiology of zoonotic diseases. Her title is Advances and Perspective on the Epidemiology of Blastocystis in Food Animals.
Thank you. Buenas tardes. Good afternoon, everybody. My goal now is to keep everybody awake. And I'm going to be changing gears a little bit. First of all, I want to thank you, the organizers, for giving me the opportunity to be presenting this data about blastocystis in animals. Okay. Okay. I don't think blastocystis needs an introduction in this audience, but I want to tell you why we care in the U.S. Department of Agriculture about working with blastocystis. And the reason is that blastocystis, you all know, is a very common enteric protist, and it has an estimated 1 billion humans infected worldwide. So it looks like it is a parasite that is worthy to study. The prevalence are really high, and we find Prevalence that are high not only in developing countries, but also in developed countries, which is also very interesting. The pathogenicity, as we have been listening over and over again this morning, is very controversial with symptomatic and asymptomatic patients. Gastrointestinal symptoms are the majority of the symptomatology that we see when we see symptomatology related to blastocystis. It has been linked to the irritable bowel syndrome and also to skin disorders, as it was shown this morning. But there is other studies that consider that the presence of blastocystis in the microbiome is actually a good sign. So there is still a lot of things we have to learn. So in my laboratory, our work, our mission is to understand protozoan parasites that are important for public health. And blastocystis is a parasite that we can find not only in humans, but in a very wide range of hosts. We have mammals, birds, reptiles, and even insects, and it was mentioned with the cockroach this morning. As a person that has been working with Cryptosporidium giardia for many years, our main challenge was that under the same morphology, we have a lot of different species, genotypes, or subtypes. So not understanding and putting the right names was actually hiding the importance of some of these subtypes. So for blastocystis right now published with the last report from the Chinese, there is 22 subtypes, with 10 of them already being reported in humans and in animals. So the potential for zoonotic is there. The transmission is a fecal-oral route, so we are going to have the potential to have food and waterborne transmission. And it's going to be transmitted through cysts that are going to contaminate food or water, also by direct contact with a host that is infected. There has been not that many, but several studies that have found blastocystis in sewage samples, in river waters. It has been a study in leafy greens, some of these pre-packed salads. They were able to find blastocystis in Italy. Very recently, there was a report that they linked a human patient with ST1 to the presence of the same ST in water for a fountain. So they did the sequencing for the water in the fountain and the sequencing for the patient, and they were able to find the same ST. And actually, they treated that patient with probiotics. So moving to blastocystis in food animals, we really don't know anything about the pathogenicity of blastocystis in animals. It's still unknown. So we don't know if it has any direct cause for the farmers when the animals are infected. We don't know if the animals are not going to gain the same weight just because they are infected with blastocystis. And the pathology, I don't think there is really any papers that are dealing with pathology right now in any of the animals that we work with. So the main reason that we work with in this public health is because it's a zoonosis. It's going to be excreted with the feces, so with the runoff, we are going to have a lot of those cysts that are going to contaminate the water. And if we are using that water for irrigation, or even for treating like fruit trees, when we do pesticide treatment, we have the possibility of contaminated food that is not cooked. So that will be a good source of infection. Here is try to let you know how we start working in blastocystis. So this, is going to be many different stories. Some stuff has been published, and some other is a work in progress, another being reevaluated because there is new technologies that we can use. But here, we used to do a lot of fecal samples looking for Cryptosporidium giardia. We have an excellent tool to look for Cryptosporidium giardia, that is the merifluor. But it's so good that you forget about looking under the bright field microscopy. So when you look, you are going to see beautiful giardia and Cryptosporidium, but you forget about what else is in that sample. So when you look by bright field microscopy, and I put an arrow in pink just in case I couldn't point it right, you can see some blastocystis cysts in that particular sample. The same applies when you use some specific antibody to detect blastocystis. You are going to be detecting beautiful cysts of blastocystis, but you are going to be missing maybe a lot of Cryptosporidium giardia or other pathogens that are present in that sample. So I don't think we need to mention in that crowd, but sometimes when we are trying to talk about symptomatology, we are forgetting that samples don't come with a single parasite. And we are all parasitologists, so they don't come just with a single parasite. They come also with viruses, with bacteria, with different diets. So there is many other things we need to have into account to resolve the puzzle. So when we started working with blastocystis, it was very challenging. We were used to use primers that were very specific and very sensitive. So whenever we have a nice amplicon band, we did sequencing, and we were able to put a name. So that was perfect. When we started working with blastocystis, that was not the case. So we have a lot of mixed banding using the primers that were currently available at that time. And in a lot of cases, even with the bands look good, when we did the sequencing, it was not blastocystis. So when you are trying to screen a huge number of samples, that is a big liability for a lab. So we tried to look for primers that were a little bit more sensitive and specific, at least in our hands, using our samples. I tried, but I couldn't get it to work. I try again. If not, I go back. So we didn't want to reinvent the wheel, so we wanted to look for primers that will be targeting an area in the small subunit region, because that's where all the STs and all the information was available in the gene bank. We tried many different sets of primers, and at the end, the one that was working better in our hands, we compared with all the other primers that were available at that time. And what you can see is our first controls. We have some calves and some pigs that we have been seeing cysts in the feces, so we knew they were positive. We didn't have at that point, another good positive control. We were not working with it. We were just starting. And we ordered some DNA from the ATCC that is trained. So we compared the primers that we were developing with the primers that were available. We looked if they were more sensitive, if they were giving us more specificity. And in most cases, when we were finding a band and we sequenced, we were getting a sequence that was blastocystis. So for us, that was a big, a big win. But now we needed to move to real samples because those have only a certain small number of STs. So we look for different animals and different sources in order to try to expand the number of STs that were going to be amplified with those primers. Because one of the problems we can have is that maybe we were selecting specific primers that were going to miss others. And if we were studying new hosts, we were going to end up using novel STs just because they were not able to amplify. So we got samples from pigs, humans, cattle, some birds, and some primates. And these are the results. We were able, in the primates and in the humans, we got ST1, 2, 3, and 4. We have 6 and 7 in the birds, ST10 in cattle, and in the pigs, we always have ST5 in that set of primers. But one of the things that was a little bit frustrating, like you think you are getting some primers that are giving you a nice band. Most of the times you get, blastocystis sequence, but we were finding in a lot of cases a sequence that we knew they were blastocystis, that they have areas that I marked in green that they were very good, and you blast them, they were blastocystis, but we were having regions where you couldn't tell what nucleotide you have right there. So the changes were so big that there is no way you can do any guessing or nothing you can really report. So at that particular time, we didn't have available a lot of the new technologies that are available right now. So cloning was the only way we have to go around and try to figure it out. So for all the samples that we thought there was mixed infections, we did cloning. And we picked 8 to 12 clones for each of the samples. And I just want to make a stop here and tell you that when we see that, it doesn't really mean that we have a mixed infection with different STs, because there is a lot of variability within the STs. So sometimes, even within the same ST, we don't get good sequence, and later we are able by cloning to determine like 6 or 7 sequences. So every time we see something like this, we cannot jump into the conclusion that we have multiple STs. And this was one of the first big set of samples that we were trying to sort out using those samples, and that data was never published. But it was 58 children that they were collecting in Bogota, and they were kids that have diarrhea. And what we were finding in those samples was mainly ST1, ST2, and ST3, like Juan David mentioned this morning. The prevalence was relatively high, it was 40%. And the STs, you can have the numbers of how many for each of the STs. But what I thought was really interesting is like in 34% of those samples, we needed to do cloning in order to really put a ST name in that particular sample. And only in one of them, in the sample number 5, we actually have 2 STs in that sample. It doesn't mean that the others didn't have it. Maybe we didn't have the resolution in order to see all the subtypes. But you can see that, for example, in number 4, we have 7 different sequences, but they all belong to the ST2. And now we move to cattle, that is where we have done most of the work. So the first study we did in cattle, we went to a farm that I can see from my window. That's easy. So we collected 47 samples. We didn't want to focus just in one age group. So we got samples from neonatal calves all the way to milking cows. And all of these animals were healthy. And we have very good records in the farm, and none of these animals were having any issues with growth or anything like that. And one of the things we wanted to know also is Blastocystis only there, or what other parasites are present there? For Blastocystis, that is what I'm going to be presenting, we did immunofluorescent microscopy, and we also did PCR and sequencing. Here, just to give you an idea about the prevalence of the 4 parasites that we look, we had that Giardia was present in almost half of the samples. It makes sense because a big chunk of the samples were post-weans, and in that age range is usually when we see the peaks for Giardia. Enterocytosome bianusia have a 36%, and Cryptosporidium at 21%. In none of the crypto we were able to find Cryptosporidium parvum. All of them were cattle-related Cryptosporidium. And Blastocystis has a 20%. And here is all the numbers, just in case you have any curiosity. I have put a line to separate the pre-weans that are all the way to 2 months. We couldn't find any Blastocystis in the pre-wean calves. In the post-wean calves with the animals in which we were able to find a higher number of positives, and we only found 1 positive in the adult cows. When you look at the number of positives, when we compare IFA and PCR, PCR was more sensitive, but there is one sample, sample number 1, where IFA was positive. Every time we have a discrepancy, we repeat it 3 times, and we couldn't get that PCR to work. But we saw the cysts in the feces. We have 3 other samples where we couldn't see any cysts, but we had positive by PCR and even able to sequence. All the STs that we find in these cows was ST10 and ST14. And this was the first time that ST14 was reported. Here is the phylogenetic tree that we developed at that time. And you can see how the ST14 clustered with the ST12, ST5, and the ST13. Here you can see the genetic distance that we have within the ST14 and also the ST14 in comparison to the other STs. And this is one of the big studies that we have been doing in the lab. And Dr. Jenny Malonis is a postdoc in my lab, and she has been really taking the lead and putting a huge amount of time and effort to make that project work. And she will be presenting the next step from where I'm presenting right now tomorrow. So I hope you come and listen to what she has to say, because it's very interesting. But just to give you an overview, we collaborated with APHIS, is the Animal and Plant Health Inspection Service. They have some monitoring that they do every 4 years for their calves. So they were collecting samples for pre-wean calves, less than 2 months. We collected 2,539 samples from 13 states. So we have California, Colorado, Iowa, Michigan, Minnesota, Missouri, New York, Ohio, Pennsylvania, Virginia, Vermont, Washington, and Wisconsin. And we didn't choose those states randomly. They are the main dairy producers in the US. So that's why they were selected. We did for every single one of them concentration of the parasite forms. In all of them with the DNA extraction. We use the DNA tissue kit, but it's true that we concentrate. We use 15 grams of feces, so we are able to concentrate the parasites and increase the sensitivity by increasing the sample. We tested for the presence of blastocystis by PCR. We sequenced, and every time we saw anything that we couldn't read in the chromatograms, we did cloning. So here is the geographic distribution in the samples that were collected in the US. You can see that we have positive calves in 10 out of the 13 states that were collected. And some of the states that were negative are the states where we collected less samples. The number of positives was low. We had 3%, 73 positives. But as we saw in the previous study, when we had pre-wean, post-wean, and adults, we couldn't find anything in any of the pre-weans. So maybe we have some kind of correlation with age. So here you can see the prevalence. We have the lowest prevalence among the positive states was in Colorado with a 0.5, and the highest was in Vermont with almost a 26%. When we look for the subtype distribution, we were actually very surprised because every sample we have looked before, we always find the ST10 and the ST14. So we were very surprised to find ST3, ST4, ST5. We did not only find like ST10 and ST14, but also ST17, 21, and 4 subtypes that we believe they are new. I have put in with a red square all the subtypes that are considered to be zoonotic, and you can see that ST4 and ST5 were also the most prevalent STs that we were finding in cattle, followed by ST14 and ST26. There was one sample that even after repetitive cloning, we were not able to get a good sequence. It was interesting to find ST17 because until that point, there has only been one report of ST17 in a rodent in Africa, and the same with the ST21 that was reported only in China in a water bag. So it looks like we have it in cattle in the US too. Also, I would like to make a comment because maybe I will forget later, but sometimes when we find some of these unusual subtypes, maybe they are not really colonizing or infecting the animal. They can be passing through. So when we are collecting a single sample and we are not following those animals or those patients for a big amount of time, they can be just passing through. It doesn't mean they are actually colonizing the gut. Just to tell you our surprise is that the zoonotic subtypes were identified in 67% of the calves that were positive to blastocystis. If you have curiosity, here are all the subtypes in the different states, and you can see how the zoonotic subtypes are very widely distributed, as well as the others. So out of these 73 samples, we have mixed infection in 15 of them. Only in 3, we were able to confirm that there was more than 2 subtypes present. And in 12 of them, we were only able to identify 1 subtype, even if more than 1 sequence for that subtype was present in that particular sample. So most of the studies has been like, you go to a farm, you collect the sample, and you get some data. So in order to go a step further, we need to try to look in the longitudinal studies, try to see if those subtypes were passing through or we see those subtypes over time. So we collect samples from the farm that we have by us. We examined 30 calves, and we followed those calves for 2 years. And we were collecting samples weekly when they were little, because there is more changes you are going to see in pre-wean calves. So we have 8 samples from the first 8 weeks. We moved to biweekly. It's a lot of work to collect 30 samples and process for 2 years. So we moved to biweekly when they were 3 to 5 months, and after 6 months, we were collecting monthly samples. But the total number of samples, even with the monthly and biweekly, were almost 1,000 samples, 990 samples. And I'm not going to bore you with all the data, specific data for prevalence in any of the different points. But what I want to show you is the cumulative prevalence that I think is something that we don't see in most of the studies, either in humans or either in animals. And the cumulative prevalence with the animals were already 2 months. It was already 50%, meaning that at least one of the samples for those animals was positive once. So we have 15 animals that by the age of 2 months, they have already have a positive sample of blastocystis. By the time we reached 6 months, the percentage was 80%. By 12 months, 97. By 18 months, all of them have had positive samples of blastocystis. In the longitudinal prevalence study, the amount of mixed infections was even worse. It's probably because we are including adults where all the ruminant flora is also voyage. So if you are looking at ruminants, the amount of protests that you are going to find in the rumen is huge. So that could be one of the reasons, or maybe because they have more time to be exposed, and we are seeing the consequences of that continuous exposure from the environment. But in 238 of the samples, we have mixed infections, which is 57% of the positives. There was a lot of samples that were untypable, even using cloning, and they were very challenging, and they become even more challenging after the animals were 12 months. So we took a break there, and we are reevaluating how we are going to continue. So the data you have in the bottom is only for the first 12 months, where all the samples were actually sequenced and cloned, and now we are going to take another approach and repeat all the samples using new technology. But the most common subtypes that we found in that farm in the longitudinal study was ST10 and ST14, and you can see that there was 80% cumulative prevalence for ST10 and 50% for ST14. We found one ST5. And now we move. So I can wake up everybody. So we did some work with pigs. Pigs was one of the first samples that we start seeing actually blastocystis in the feces. When we have a visiting scientist, she really had a passion, and she was always looking if blastocystis was present. So because we have the capability of go to a farm and collect the samples, there was a colleague that was doing a study with pigs, and we asked if we can collect some samples and look if they have blastocystis. So we collected the 28 samples that were in her experiment, and all of them were pigs that were 5 months of age. We collect when they were going to do the necropsy. All of them were positive. A lot of them require cloning, 57%. And the STs that we found was ST1, 3, and 5. You can see that the correlation between IFA and PCR in the pigs at 5 months was clear, a very good correlation. But as I mentioned before, only in a few cases we were able to see 2 different subtypes, and in most cases we were seeing multiple sequences for the same subtype. So after we did that, we were trying to go a little bit farther and try to figure it out where blastocystis was in the gut. So that was another step. There was some animals from the same group that we collected before that they were going to be doing necropsy. So we checked the feces before, and we selected the 5 pigs that has the higher number of oocysts in the feces. And for them, we collect not only the segments to do histology, we also collected the content to see if blastocystis was in the lumen of the different sections of the gut. Only one ST was identified, ST5 in all of them. What makes sense because it's the same farm, and that was the only ST that we have found previously. What we found is that also makes sense is that all the samples from the cecum in the lumen, we were able to identify blastocystis. When we look in the sections that we did DNA extraction from the sections as well as microscopy, we were able to find positives for the jejunum, the colon, and some of the cecum. And here's some of the histological sections. You have A and B is the colon, C is the cecum, and D is the jejunum. And the A and the B, we stain it with the host radius peroxidase and hematoxylin, and blastocystis will stain in brown. And in the other ones, we stain with parafluor and DAPI, and you are going to see blastocystis as green. And we saw that when we did histology, we found that the proximal and the distal colon was the sections of the gut that were more heavily infected. And also we found that blastocystis was attached to the feces in most of the cases. And only in the C, we were able to see the blastocystis attaches to the epithelium of the intestine as well as to some feces. And also, I didn't put it here, but we didn't see any lesions associated with the presence of blastocystis. And we move to the chickens. And this is just a very little study that I have done with a visiting scientist from Brazil. She was doing a PhD sandwich and did a portion of her research in our laboratory. And blastocystis was not in her original plants, but since we have the primers and the capability, we decided to go for it too. So this is the first genetic characterization of blastocystis in chickens in Brazil. Maria Julia collected 130 chicken samples from 17 different markets in Uberlandia and Belo Horizonte in the state of Minas Gerais. And all the chickens were healthy, and they were between 1 and 1 year. They were just chickens that were in the market. We find that the distribution, there was a wide distribution of blastocystis. The prevalence was high. It was almost 34%. 44% of the chickens were positive. And it was very wide distribution at the markets too. So we have blastocystis positives in 12 out of the 17 markets, so 71 of them. And we identify 5 subtypes. 5 that has been previously identified, 4 previously identified, and 1 identified in our lab, the ST-25, and a novel subtype that has been observed in 2 chickens, but we need to do a little bit more of research before we can actually determine if it's a novel subtype or we can put a number on it. If you have interest, these are all the data for each of the markets with the percentage of positives. You can see that although the prevalence is 34%, if you remove actually the negatives markets, you can see that the prevalence is actually quite high in most of the markets that are positive, with a prevalence that goes for 20 to all the way to 75%. The STs that we identify, mainly ST6 and ST7, the most common, 58 and 98%. Mixed infections were very common, 63.6 of them. And there was a clear co-infection of ST6 and ST7 in 45% of the samples. Intra-subtype heterogeneity was very frequently observed, especially in ST7, where we are finding very challenging to even deal with it. And Jenny is working in doing all the next generation sequencing analysis on all of these samples. So after all of these years working on all of these thousands of samples of animals, check what we have learned. We have learned a little, but we still have a long way to go. So we know that blastocystis is prevalent in our food animals. We were able to prove that they are in cattle, they are in pigs, they are in chickens, and we have other studies in different animals where we have seen it too, or even in wildlife in deer. But so the animals can actually be a reservoir for infection for humans, for other animals, wildlife. We have identified subtypes in food animals that are actually, they have the potential to be zoonotic since they have been identified in humans before. We find it very interesting to find in the pre-wing calves a lot of ST4. That is one of the main STs that is found in humans. However, as we all see, as we are doing more research, the genetic diversity of blastocystis is only increasing. The host range keeps increasing, and there is not a clear association between the STs and the host. So it looks like we are only scratching the surface. We know that the food animals may have a role in the transmission, but we still need to work a little bit more to try to understand what is the role of animals in the transmission and epidemiology. We need to use new technologies to actually like face some of the challenges that we are seeing with these samples. And I think right now we are only seeing the top of the iceberg, just scratching the surface. And I think with the development and implementation of new molecular tools like next generation sequencing, I think we are going to be able to answer a lot of the questions we are talking about today. I think it's going to help us to understand better the association between blastocystis, genetic diversity, and disease, because we need to put a name before we can actually make a connection. We are going to maybe understand better that the asymptomatic case has something else that is not what we thought it was. We are going to understand host stability and understand better the microbiota and the potential pathogenicity for the different STs. And in our case, one of the main reasons why we care is we would really want to understand the transmission dynamics and the zoonotic potential for this parasite. So with that, I would like to acknowledge a lot of people that work on these, because all of these samples cannot be processed by a single person. So, and I want to give like a special thank you to Jenny and Alexi, that are the members of my team in the lab, and they really do a lot. Ronald Fayer, that was the one that started with me the journey of blastocystis, and a lot of the students that have helped with collecting, processing, PCR sequencing samples. Teresa, that even provided a lot of the samples when we started. Gloria and Urban, that helped us with all the PIC work. Ted, that helped with a lot of the histology, and the APHIS people that did all the collection for the NAMS study. And thank you so much.
Thank you very much, Monica, for this very nice result of the epidemiology of blastocystis infection in the animals. So now it's open for discussion. Yeah, Ray?
Thank you very much, Monica. Thank you for an excellent and exciting talk. And I'm a huge fan of your work, and I think it's so inspiring to see all your data, all your observations, and your massive sampling, and all the things that you do, and not only with blastocystis, but also with cryptosporidium and all of those things. So where to start? I was just wondering, you had this massive studies of the young calves. So how many of those had actually also cryptosporidium, for instance?
Cryptosporidium, in those pre-weaned calves, we have a prevalence of around 40%.
40.
I think now when we finish all the next generation sequencing, I think we can try to put a paper together, because we have looked in all of those samples, we have looked for Giardia, we have data for crypto, and we are looking for Baianusi too.
Because I was thinking that maybe in the older cattle you might see a higher prevalence and a more sort of sustained subtype distribution with subtype 10 and 14, and that's also what you showed. So the prevalence was much lower in at least some of the calves, and they had a much more random subtype distribution. So a lot of things could be interfering with that. Also, you showed, and that might be quite confusing to some of the members of the audience here, that you had a lot of other subtypes as well. And usually we say that until now we have found 17 subtypes in humans and non-human primates and mammals and birds, but you have additional subtypes as well. Have they been confirmed by like full gene phylogenetic analysis?
I don't know. The 18 to the 22, it was published by a paper in China.
Yeah.
We were able to find some of those subtypes in our studies.
Yes.
For us, being able to find it in a place that is so far probably.
Yeah. But have they been confirmed by complete ribosomal gene analysis?
I don't think they have the full.
Yeah, so they are like tentative, potentially new subtypes that need, because we know that also that within some subtypes you really have a lot of variation. Yeah. So, for instance, subtype 10, subtype 5, subtype 7, we have a massive.
I mean, we have a lot of issues with
yes,
ST10.
Yeah. So So,
I mean, ST10, the variability is huge.
Yeah.
The problem is like we are trying to be conservative, but I
Yeah.
think the variation that we see in ST2
Yeah.
probably is enough to
Yeah.
split it, but we need to have enough samples and enough support
Yeah.
in order to put another subtype out there.
Do you have a culture? Do you cultures?
We are not doing culture.
So you don't have a subtype 5 culture, for instance, because you have a lot of subtype 5 in your pigs?
I mean, we can try, but right now we
Yes.
don't have anybody in the lab that can put the time to do some of that.
And given your zoonotic focus, is there any reason why you don't try to obtain more resolution, because you have all this subtype data, but don't you ever want to just dig one step deeper and see what is the genotype resolution here, the allele? Because, you know, when you do the barcoding, when you use the primers by Scicluna that are used by many of our colleagues, they are able to sort of step down one level and say, okay, here we have allele number 1, allele 4, like a genotyping system.
I mean, our primers overlap in most of the fragments. So even the forward primer is the same. What we changed was the reverse primers just to increase the sensitivity. When you screen that many samples, sensitivity is very important. Otherwise, you find yourself working extra hard and putting extra money and resources just to get something that is not blasto. But it is possible to go back to samples that are positive and try to look for the alleles.
Yeah.
But we are, I mean, our approach right now has been trying to go to the next generation sequence and actually trying to sort out the mixed infections. Because that's for us the main way to go a way deeper. Because if we are finding 7 different nucleotide sequencings within an ST in a particular sample, we have a lot of variation just in that particular sample. So that animal, I don't know if that will be what the alleles will be, but we have such a huge number of sequences that I don't know if we can replicate those when we use different primers. Because what you are getting with cloning, if we use different primers, that doesn't mean that even if I pick 20 clones later, I'm going to be able to get it.
Yeah.
So I think that's one of the problems that we face. It doesn't mean that maybe for particular samples where we find a novel ST, we cannot go and try to go and amplify something else. We are also trying to look for different markers, but it's challenging. It's very challenging. So we tried with the MLO that there was some papers that looked promising, but it looked very promising. We used ATCC, but as soon as we moved to fecal samples, things didn't work that well. So, I mean, we put a lot of work into it, and it didn't really pay off.
But you do get a lot of papers saying that because we found subtype 1 or 2 or 3, this in animals and in humans, this means that it's potentially zoonotic. But I think sometimes we need to get more information to see what is actually the intra-subtype variation before we can actually say that something is zoonotic.
And that's why we were trying to see if we can find another locus that is able to amplify all the different subtypes. We were able to go a step further.
Okay, good. Thank you.
Is there any questions? Go ahead.
Hi. When you were talking about blastocystis in pigs, you found 3 subtypes, 1, 3, and 5. But whenever you found mixed infections, it was always subtype 3 and one of the others. So do you think that subtype 3 has an particular characteristic that allows this to happen, or does that have an explanation?
I honestly think that ST5 is the most prevalent ST that we find in pigs. I think when you do cloning, you can end up way deeper and maybe identify some of the other subtypes. But the point that we do clone and we pick 10, 12, or 100 clones, that doesn't mean we are actually seeing everything that is going on in the sample. Because the way the PCR and cloning work, we are just picking. So it's just pure luck that you are picking some that are different. So that's why, we pick 8 to 12, but that doesn't represent all the different sequences and subtypes that you maybe have there. That's why we want to move to another technology that is going to go deeper, and we are going to be able to find all those subtypes that are a minority within the sample. Because that has been a problem also for Cryptosporidium in water samples. Like we have a great PCR, you do sequencing and cloning, and at the end what you find is whatever is the majority. So if we keep picking up clones, we may be ending up finding another ST. But it's just a matter of how much effort you are going to put into cloning and how much samples you are willing to process.
Thank you.
Is there a question, comment? Nothing? Okay.
Hi, Monica. Thank you very much for delivering such a nice talk. Any chance that you might include human samples from in this kind of studies? Because I think you are in a fantastic position to try to demonstrate zoonotic transmission. So with the technology that you have, and if you can include human samples and can demonstrate that they are infected by the same subtypes, that would be fantastic.
It's very hard for us because we are an agriculture laboratory, so we are not allowed actually to go and collect. The samples that I have for humans was through collaboration with others. But we are willing to help and to use whatever we develop in order to try to unravel, because that is our mission. And I think one of the reasons of coming here is try to get to know other people, try to see what other people is doing, and try to see between all of us we can find a better way to understand a little bit better blastocystis. So, I mean, anybody that has any question or samples in our lab, we are always happy to help or to do some of the PCRs, cloning, and try to figure it out among all of us. I mean, research is about collaborating, so we have to do it as a team. Otherwise, I don't think we are going to go very deep with this.
Don't worry about that, we will. Thank you.
Is there any questions? So my turn.
Thank you.
So in the survey on the United States, in the cattle, it looks like on the more high population states, it's more high number of the different subtypes in the slide.
It is hard to make a big statement because, in order for, we pick the states based on the states that are higher producers. But later, the farmers have to agree to collaborate and let us go into the farms to get the samples. So we don't know, there is a confidentiality also, so we don't know where the samples are located. The only access that we have for data from the farm is how big is the farm. So we have divided the farms between small, medium, and large. But because the prevalence was small, we didn't have that many positives. It's very hard to...
I'm wondering of some of the, how we can transmit it from the many kind of the subtypes to cattle. Maybe some of the other human or animal to exploit it of the fecal samples to contaminate it in this area. So this is originally come from more, different of the animals or high population of humans is more high possibility to other lower population of peoples.
It could be where they are located, and one of the things with the pre-weans is that in pre-weans, their gastrointestinal system is like us. They are still monogastric. So the rumen is not going to develop until they are post-weans. So maybe in pre-weans, we are seeing all of these subtypes that we are finding in humans just because their gastrointestinal tract is more similar before they reach the 2 months than later. And that has been something that has been shown, for example, for Cryptosporidium with parvon.
Yeah. And how do you think about after the growing up of the cattle, finally 2 years later, it's 100% positive?
What we are seeing is that pretty much after they reach the 12 months, pretty much every other sample is positive. So we can see that at the beginning, the animals that are 1 or 2 weeks, I think in the first 2 or 3 weeks, we only have a couple of samples that were positive. If you move to the 18 months, pretty much all of them are positive. So I think it sustains in the time. But maybe ST10 and ST14 have a more prevalence there.
Because the fecal oral transmission is very easy to, if this parasite already adapt on the host animals. So if your target animal from the young age to the 2 years later, this animal will be growing in the same gauge or same field or CIM farms should be positive rate is increasing in the term of the.
Yeah, and that's why we say like after 12 months it was becoming really challenging.
Yeah.
And I think the point is that, you know, if you go to any barn where they have milking cows, they are all together, they are sharing a lot of spaces, they share a space with humans. I mean, you hope they don't have rodents, but there can be other, you know, animals and birds that can be passing through. So that's why if you find an unusual, I'm having my reservations to say that that is actually infecting the animal and not just passing through.
Yeah.
But we are finding that the population or the diversity that we are seeing in the older animals is much more higher, and it's probably because they keep picking up different things from the environment. And if it's not an infection that has, you know, we don't know what is the pre-patent and patent period, so it looks like whenever they get infected forever. So they keep adding things from the environment, and at the end we have really complicated samples when the animals were getting older.
Well, congrats on your work. I think it's amazing that you work with agricultural, like that kind of field. I just wanted to ask, in the cows population, you saw that the little cows, they didn't have any positive samples, right?
Yeah, the prevalence that we see in the pre-weans is much more lower.
Okay, because in pigs you use 5 months. Every pig has the same age.
We
Do you
use
think
we
that
use
would
those
influence?
when they were at necropsy, so that's when we got the
Oh,
samples.
okay.
But actually, I don't have the data, but we did a longitudinal study in pigs where we start collecting the pigs the week they were born, and we follow them until they were 5 weeks. But I haven't had time to really go through all of those samples. But we see pigs that are already infected with blastocystis at week 1. Most of the subtypes that we found, it was mostly ST5, but we haven't completed all the study, but we follow the pigs since week 1 until they were 6 months, but we don't have that data yet.
Okay, and for example, in chickens, did you see any difference between ages?
No, these chickens, the number of positives was not big enough to really extrapolate and make big conclusions. The problem is, like, in order to make big statements, you need big numbers, and sometimes it's hard to get that sample size. So hopefully, you know, we will be able to, you know, explore more, be able to get more samples, have samples also for other geographical locations, because if you look in birds, there is actually not really that much. I mean, Colombia has a great bird population, so maybe the presence of the ST6, ST7 in humans is related to the birds, but we need to do more studies in order to be able how we can put all the data together. But we need a lot of samples in order to make big statements. But with cattle, I feel pretty confident saying that there is really an association with age, and in the pre-wean calves, you have less.
Thank you.
Okay. So no questions anymore? So thank you very much, Monica.
Thank you. And our next invited speaker, I don't think he needs an introduction, is Hisao Yoshikawa. He is from Japan, and he's going to talk about blastocystis and zoonotic potential. So please.
Okay, thank you, Monica. So first of all, I need some of the corrections in my abstract. So please delete our exchange after my final presentations. Zoonotic potential of blastocystis parasite has been considered based on the homology of the SSUR DNA sequences between humans and animal blastocystis organisms, and higher prevalence rate of blastocystis infection in animal handlers compared to the ordinary people. In this session, we report the blastocystis infection among 10 local families where fecal-oral transmission of various parasites between humans and animals have likely been common. If blastocystis is zoonotic parasite, the resident people and their domestic animals will share the same blastocystis colonization. For this purpose, we have visited Sumba Island in Indonesia many times. It's about 1 hour away from Bali. The service area is located at Wanyap, East New Tenggara province, where has been the endemic area of malaria parasite as well. Resident peoples have been living in these traditional open houses. They have been using rain, river, and well waters for daily life, and there is no toilet in most houses. This situation is highly risky for the transmission of various parasites between human to human or animal to animal and human to animals via fecal-oral route. Indeed, high prevalence rate of several common fecal-oral transmitted parasites were observed when direct smear examinations was performed by light microscopy. In the resident houses, pigs, chickens, and buffaloes were common domestic animals, while goats were rare animals. Among various animals in this slide, monkeys are the most infected animals with the same subtypes with humans. However, there is no monkeys in our service area. As shown in the previous slide, the resident peoples have many pigs, chickens, and buffaloes, while goats are rare in the service area. So pigs and chickens are good targets for this research because these animals are also shared the same subtypes with humans. This scheme is a life cycle of Blastocystis parasite in the survey area. Surrounding of the houses is contaminated with human and animal feces. Hence, humans and animals are frequently transmitted various Blastocystis subtypes each other via fecal-oral route. If Blastocystis is capable of colonization various animals, including humans, the same Blastocystis parasite should be detected from humans and animals. To clarify the zoonotic potential of Blastocystis parasite within its life cycle in the endemic area, we collected fecal DNAs from residents and their domestic animals among 10 local families. Then we judged the levels of Blastocystis colonizations in the samples based on the PCR amplification. If the same levels of colonization of the same subtypes were detected, sequencing of the SSUR DNA was conducted, and nucleotide homology between human and animal origins were compared for evaluation of zoonotic potential. These 10 families around the school were randomly selected, and the fecal samples were collected from them and their domestic animals, as many as possible. A total of 48 of human fecal samples were obtained from 10 families. In chickens, we could purchase chickens from 4 families and used cecum contents for this study because sampling from fresh fecal sample from chickens is usually difficult. So we could not collect them from 4 families in this slide. Pigs and buffaloes are common animals in the residence, while only one family had goats. Fecal DNA were extracted by ZR fecal DNA miniplex kit because this kit has been superior to the other common kit as described previously. Extraction was repeated 3 times from the same columns in the final step. These 3 DNA samples obtained from each fecal sample were used for PCR amplification for evaluation of DNA content of the fecal samples. For evaluation of colonization of Blastocystis parasites and identification of Blastocystis subtypes, 9 sets of subtype-specific primer pairs developed from the unique sequence of the SSR DNA were used as reported previously. This method can also detect mixed subtypes in the same samples. Since the extraction volume and PCR conditions for the 9 sets of the primers were the same, colonization of the target subtypes was evaluated by the band intensity of the PCR amplicons from the first to third extracted DNAs. This slide shows the Blastocystis colonizations, non-colonization, and negative amplification among human and other animals. Based on the band profiles of PCR amplicons, 19 samples of the resident were judged as Blastocystis colonizations, while 10 samples were judged as non-colonizations because these DNA samples showed faint band profile in the first extracted DNAs and no band profile in the second and third DNAs. Other 19 samples were totally negative for Blastocystis. In chickens, 8 samples showed Blastocystis colonizations, while 3 samples were judged as non-colonizations, and 3 others as PCR negative. In pigs, most of the samples were judged as Blastocystis colonizations. All of goat samples were judged as Blastocystis colonizations, while all of buffalo samples showed non-colonization or PCR negative. In resident, all 9 subtypes could be detected by PCR amplifications. However, Blastocystis colonizations were only subtypes 1 to 3. Blastocystis colonizations in chickens were only observed in subtypes 6 and 7, and Blastocystis colonization was not observed in the buffaloes. In humans, non-colonization of the subtype 6 and 7 were also observed. I will discuss this issue later. In pigs and goats, all PCR positive samples showed intense band profiles, suggesting Blastocystis colonizations in these animals. Interestingly, faint band profiles were not observed in both animals. These colonizations were subtype 5 in pigs, while in goats, mixed infection of subtype 1 and 5 were observed. Since the colonization of subtype 1 was also detected in the resident, sequencing of the PCR product was conducted to clarify whether both subtype 1 parasites are the same or not. Sequencing of the PCR amplicons amplified by subtype-specific primers were conducted by cloning method. Since multi-copy genes have been known in the target gene of blastocystis, 5 randomly selected clones were sequenced and tested whether 5 clones are sufficient to reveal diversity among multi-copy genes. Then nucleotide sequences of the variable region of the genes were compared between human and animals. The variable regions has been known to be useful for identification of 9 subtypes of human blastocystis. Furthermore, it can be easily distinguished into subgroups within the same subtypes as reported previously. For this purpose, we tested a reference blastocystis subtype 7b strain because this strain has accomplished genome sequence and 17 multi-copy genes of the SSU RNA appeared in the gene bank. As shown in this slide, alignment of the variable region of the copy genes can be categorized into 8 different nucleotide sequences. Nucleotide sequence among the copy genes was up to 40 bases. Based on the gene bank data, these variations of nucleotide sequence in the variable region were observed among the subtype 7. As shown in this slide, all copy genes of the reference strains are categorized into the subtype 7A subgroup. These 3 subgroups are based on the phylogenetic tree inferred with entire sequences of the gene as shown in the next slide. These 3 subgroups are clearly separated into 3 branches in the tree inferred with the entire sequence of the gene. Although up to 40 basis differences were observed among the copy genes, all copy genes were categorized into the subgroup A in the variable regions. Therefore, phylogenetic positions among the copy genes was constantly the same in the phylogenetic tree. So we had tested whether sequencing of 5 clones in each PCR product is enough for detection of the diversity of the copy genes. When we had amplified the reference strain of the subtype 7 with our developed subtype 7 specific primer pair, followed by sequencing 5 randomly selected clones, these 4 different nucleotide sequences highlighted by yellow color were detected. We could not detect single copy genes among 5 randomly selected clones, but we could detect all sequences of the multi-copy genes of the same nucleotide sequences in this case. Therefore, random selection of 5 clones from the PCR product may be enough for screening of diversity among multi-copy genes, as well as detection of mixed infection of different blastocystis parasites among the same subtype. This is the phylogenetic tree inferred with complete sequence of the SSUR DNA of all 44 data of subtype 1 in gene bank and other subtypes. As all subtype 1 data in gene bank are clearly separated into 3 subgroups with high BP values and named as A to C. These 3 subgroups also showed distinct different nucleotide sequences in the variable region. When first and second extracted DNAs were used for PCR using the subtype 1 specific primer, several human and goat samples showed intense positive band profiles in both the first and second DNAs. Therefore, sequencing of the subtype 1 Blastocystis organisms were conducted to examine whether subtype 1 Blastocystis of humans are the same or not with those of the goat Blastocystis subtype 1 organisms. As described previously, we examined 5 randomly selected clones of the subtype 1 amplicons of humans and goat DNA samples respectively, and then compared the nucleotide sequences of the variable regions. This is the result of alignment of a total of 70 clones of subtype 1 amplicons obtained from 10 humans and 4 goats fecal samples. As you can see in the upper figure, except for 1 clone from human DNA, 4 clones from humans and 2 clones from goats were completely same sequence of the subtype 1A. Therefore, Blastocystis organisms of subtype 1A from humans and goat must be same origins. Likewise, majority of goat clones were the same with the human subtype 1C nucleotide sequences. This is summarized haplotype distributions among humans and goats based on the nucleotide sequencing of the variable regions. Interestingly, mixed haplotypes could not detect from human samples, while 2 out of 4 goat samples showed mixed haplotypes of A and C. Based on the PCR amplifications, colonization of subtype 1 organisms was evident in both humans and goats. However, all 5 clones from the same humans were constantly the same haplotype, and 2 different haplotypes of A and C were detected from human samples. In contrast, 2 out of 4 goat samples showed mixed haplotypes. If human infection of the subtype 1 was originated from the contamination of goat fecal materials, some of humans should be mixed infection of the haplotype A and C. In this study, however, we could not detect mixed haplotypes from the resident peoples, suggesting the human infection of the subtype 1 might be originated from the contamination of human fecal sample materials. The mixed haplotype infection of goat samples, suggesting that the origins of the subtype 1A and C are human fecal contamination. In goat, colonization of subtype 5 was also detected. The subtype 5 is also major blastocysts in pigs. In the resident, non-colonization infection of the subtype 5 was also observed, and subtype 5 and 6 were major subtypes of the non-colonization blastocysts infection in humans. In humans, colonization of subtype 1 to 3 is evident, while non-colonization levels of these subtypes was also observed. Therefore, it is important to clarify whether non-colonization levels of human subtype 5 is derived from pigs and goats or not. This is example of PCR result of the subtype 5 amplifications. As a result of PCR amplicons, using the first and second extracted DNAs, colonization of the subtype 5 was evident in the pigs and goats, while minor amplicons were only observed first extracted DNA samples from some resident. Therefore, we compared nucleotide sequence of the variable region of the target gene among pigs, goats, and humans. These two subgroups, A and B, showed distinct different nucleotide sequence in the variable region of subtype 5. Sorry, I mistake. Based on the phylogenetic tree, all 9 data of subtype 5 in gene bank were separated into 2 subgroups with 100 BP supported and named as A and B. These 2 subgroups, A and B, show distinct different nucleotide sequence in the variable regions. Based on the nucleotide sequences, all 60 clones from 12 subtype 5 positive pig samples were categorized into subtype GO-B. In the goat samples, all 25 clones showed the same nucleotide sequence of the subtype GO-A. In human samples, both haplotypes, subtype GO-A and B, were detected. The same nucleotide sequences of the majority of the both haplotypes were also observed in gene bank data. These nucleotide sequences might be the dominant copy genes among the subtype GO-A and B Blastocystis populations. Surprisingly, pigs and goats showed clearly different haplotypes, while in humans, various levels of the mixed haplotypes were observed. These results suggest that minor infection of the both subtype 4 haplotypes in the resident is originated from the contamination of the both animals. Blastocystis colonization in chickens was subtype 6 and 7, while the subtype 6 was frequently detected in humans as non-colonized level. Therefore, sequencing of the subtype 6 amplicons of the chickens and humans were conducted and nucleotide sequences compared among both origins. All 20 subtypes 6 data in gene bank were not separated into different branches. Accordingly, alignment of the variable regions of all the 20 data showed minor difference of the nucleotide sequence. When cloning of the subtype 6 amplicons was accomplished, A total of 26 clones from 2 humans and 4 chickens could be sequenced. Although the forward primer was developed from the variable regions, the remaining part of the variable regions suggests that most of the human origins are the same nucleotide sequence of chickens. Therefore, non-colonization infection of the humans might be originated from contamination of the chicken fecal materials. In summary, fecal contamination between human and domestic animals were frequently occurred in the survey area. Hence, various Blastocystis subtypes were transmitting between each other. However, only colonization of subtype 1A and C were observed among humans and goats, suggesting subtype 1A and C are zoonotic Blastocystis parasite. So please delete the description of the zoonotic parasite of the subtype 5 in the abstract. Furthermore, in this study, 2 genetically different subtype 5 Blastocystis populations were appeared in pigs and goats. The finding of the host-based restriction of genetically different populations within the same subtypes is the first report. Therefore, subtype identification is not enough information to support the genetically similar or same organisms based on the subtype classification. It is essential to show detailed molecular evidence for evaluation of zoonotic potential of this parasite. Molecular sequencing of the variable region may be one of the reliable approach for this issue. This work supported of the CACAIN grant number. So CACAIN is Japanese government supported. So thank you attention. So this place also, Japan has many beautiful places of the castle, especially in the April is quite beautiful. Thank you.
Thank you very much for your great presentation. Please, there is any questions? Any questions for the audience?
That was really interesting. I'm wondering, how easy is it for you to assess what is colonization versus non-colonization? Is it just how faint the bands are on the gel? And
yes, that's right.
Is it a continuum of faintness, or is it
only
usually
the faint band of the first destructed DNA, extracted DNAs, and second and third is no band anymore. This is a category.
I see. Okay.
But the colonization levels, we can get to the very thick band of the first, second, third sometimes. So we judged of this real colonizations. So it's quite easy to distinguish. Yeah.
Thank you.
Thank you.
Any other questions?
Oh, here.
I have one question.
Yeah.
Did you collect more than one sample in each of the humans and the animals, or it was just a single sample?
This is only single sample.
Only single
yes.
sample. And one more. You are finding the ST1A and ST1C in goats and in humans. Were those goats and humans from the same family? Because it would be interesting to see the different subtypes that are associated with a nuclear family. So are the subtypes shared between the family members and their animals more than just seeing the
Subtype
picture?
1. You're asking on subtype 1?
Yeah, the ST1A, 1C we're finding humans and animals.
Yes.
So
You can see on the human cases, it's all of the only one, and then family 4 has one person has different subtype A.
So they come from the same family.
Yeah.
Hi. I wanted to ask, and maybe you said this and I missed it, but the A, Bs, and Cs that you were assigning within subtype, how different are those sequences in terms of sequence?
I'm not calling the subtype for A, B, C.
Right.
Subgroups, okay?
So the subgroups, how different are those subgroups in terms of sequence similarity?
It's almost nearly 100% of BP. Here. For example, subtype 1, between the A to B, 88, 96 or 99 supported. In the subtype 5, in case it's 100% BP.
But those are like bootstrap.
Yes.
I guess I was asking, like, along the length of the sequence, how many nucleotides vary between the different?
Only few number in the variable region.
So like they're 98% similar or 99% similar, the different
it depends on the subtypes.
Different subtypes have different,
yes.
Okay.
In subtype 7, it's quite many of differences. I showed the first of slide here, even though we can easy to categorize of the ABC.
Okay, thank you.
Any other questions?
I have a question for you after my presentation.
Okay.
For these results to show of if you can use of the sequencing method for subtype identifications, all of the result of the non-colonization levels should be come out of the positive result of the sequencing. So I really want to know that the sequence method is to reflect on the real colonization or non-colonization levels. How we can judge the result to come out of positive in sequencing?
I mean, I don't know. I have a problem understanding the colonization versus not colonization just by seeing the strength of an amplicon in a PCR, because a regular PCR doesn't really quantify the amount of parasites that you have. So I think if I wanted to talk about colonization, I would like to see that parasite is present in time before I can actually say that that animal is being colonized or infected versus something that can be passing through. Mm-hmm. Because passing through is being seen. I mean, if you do wild carnivores, that I think David is going to present some of that data tomorrow with you can see different subtypes that are associated with rodents just because the mesocarnivores are going to be eating those, and doesn't mean they are colonized, and you can have a very strong band for those. Yeah. So I think we have to be cautious when we say that a person or an animal are infected or colonized. I think most of the times we don't have enough data to make that assumption, but maybe I'm very conservative. I prefer to say they are positive for blastocystis versus being colonized unless we are able to demonstrate that they are being positive for a length period of time and are being shedding in the oocysts. Because if you infect an animal with an ST that is not suitable for that animal, you are going to see some of that DNA coming through the feces. But we know it's not actually infected that. Yeah. But if we put
I'm really afraid of the new subtypes or novel subtypes is coming out of the several papers after sequencing, the short sequences. And then it's real colonized or not. So we have no way, just report it, I read. How we can judge it? This is a problem.
I think we have, when we see a new subtype or a new sequence once, and we are not able to see that sequence anymore, I think we have to be cautious.
Other information, it's really, they have plenty in the gut, in the intestines, they colonizations, this new novel subtypes. This is important evidence before we can recognize of this new novel subtype come from these different animals.
I mean, I think when you have a novel subtype that you have been able to isolate for a big number of animals, I think you feel pretty confident that that subtype is actually present in those animals all the time. Yeah. If you find it only once, I think it's almost impossible to know. Yeah. You know, the example that we put with ST-17 or ST-21, if there is only one report, and one is in rodents, one is in a water bag, and now we are finding in cattle, maybe we haven't explored enough to actually be able to point where that subtype actually belongs. So what is the major host for that subtype? I think we still have a lot of work to do in able to actually determine the zoonotic potential, or what are the major hosts for some of the STs. But I think it's very interesting to be able to see how we can break some of the STs, because ST10, ST7 is being very challenging for us because of the huge diversity that we see. So, I mean, anybody that has ideas or suggestions, you know, because at one point we are going to have to break them somehow. Yeah. I mean, even the STs, if we were working with another parasite, there will be species. We just don't have the information to call them species, and I think we shouldn't without having all the biological data. But I think under the umbrella of Blastocystis, we are putting a lot of things that are different species. Yes. So, I mean, as I mentioned at the end of my presentation, I think we are only scratching the surface. But hopefully, since the community is growing, the purpose are growing, you know, I think the next time we have a Blastocystis conference, we will have many more answers for our questions. So. Yeah. Anybody else have a question? Maybe? No? When
you say that Blastocystis can be seen as a passenger, is it a cyst or is it only like free DNA?
Pardon me? What mean? The passenger is, it depends on the phenotype. It's destroyed or still alive or still formed. It depends on the condition. Cyst is really tolerant.
Yeah, but so when you detect Blastocystis and it's not colonizing the gut, do you get the detection from only DNA or from DNA that is extracted from a passaging cyst? You know?
I mean, in my case, when I say that it's passing through, if I infect, and I'm not going to put Blastocystis since we don't understand exactly what are the major hosts, but if I look for a Cryptosporidium species that is host specific, and I put it in another animal that I know is not going to be infected, if I collect samples, fecal samples, for 7 days, in those couple of days after the infection, I'm going to actually be able to detect DNA, and in some cases, depending on the number you infected, cysts or oocysts. And that doesn't mean it was actually infecting the animal. That's why when we don't have studies that, you know, are a little bit like more long-term, it's hard to actually say they were colonizing, passing through, or, you know, or that we were lucky that day. I mean, we did a study with Microsporidia in baby calves, and we went to a farm that there were so many flies in the milk, and when we ran the microprimers, we were like, wow, this farm has a lot of Microsporidia, but most of the Microsporidia was actually insects, Microsporidia, and we were detecting that in the feces of the calves. So that's what I mean with the possibility of being passing through, especially when we are doing DNA extraction. But even if you do microscopy, you can still see the cyst, or in other parasites, the oocyst, even if it's just passing through, if it's not the right host. Oh my God, everybody's quiet. Well, if you don't have any more questions, I think we can finalize. This has been a great, productive day, and very excited research, and I'm looking forward to learn more about what you all have to say tomorrow. Thank you.
Thank you.
We deeply thank you for your participation in the second International Blastocystis Conference. This was session 1. We now declare it closed.